We're going to start our tutorial, Nanomedicine, Principles and Applications. We will be with you, Dr. Juan Vivero Escoto, and myself, Daniela Salado. So before starting, we are just to introduce ourselves. Dr. Juan, if you want to introduce yourself for the audience.
Yeah, good morning, everybody. I guess my name is Juan Vivero Escoto, as Dr. Daniela was mentioning. I'm a professor at the University of North Carolina in Charlotte. Yeah, and I'm more than excited to be here to impart this tutorial and I guess to share with you different topics that we have related to nanomedicine, and looking forward for a great tutorial.
Thank you very much. And myself, Daniela Salado, I'm coming from a national program called Cátedras CONACYT. I'm working at the University, Autonomous University of San Luis Potosí as young researcher. Also in topics related to nanomedicine, some different applications. And as Dr. Juan Vivero Escoto said, I'm also excited to be today with you. As you know, English is the official language of this international conference. So we'll be given this tutorial in English, but if you have questions and you need to ask in Spanish, we are here and with this possibility to be more clear in what we are presenting and sharing with you today. So in the screen that we are sharing with you, you can find like the general outline of today's tutorial. So we'll be talking about how we can design and synthesize nanoparticles, particularly with potential applications in medicine, some physicochemical characterization techniques that we are using, how to evaluate these properties, then how to characterize the biomedical properties of the nanoparticles, some barriers that we will find when we try to apply these materials in medicine, some toxicological aspects, some applications in drug delivery, medical imaging, immunotherapy, and how is the nowadays this transition from basic research to the clinic. So this is how is organized the tutorial. I will let you know that we will have a little break around 11:30, then we have lunchtime. And so welcome again, everybody. And if there is no comments, or well, we would like to know about you. We are a small group, but I think that we are all here with this, how to say, exciting to know more about this topic. So we would like to hear about you. You can open your microphone and introduce yourself, like what you are doing, which are your research interests. So please, if someone of you want to share with us this kind of information, it will be helpful for us in order to be more accurate in what we are presenting today.
Hello, hi everyone. Can you hear me?
Yes, we can hear you.
Hi Daniela, hi Juan, and hi everyone. My name is Eduardo Ruiz Hernandez, and I'm also an associate professor in Trinity College in Dublin. At the moment, I'm in Spain on my holidays, so it's my pleasure to connect with you and to share this tutorial. My research interest is also with drug delivery, with the synthesis of nanoparticles, and well, some of the topics that you have today are a bit outside of what I'm doing at the moment, so I'm very interested to know a bit more of the basics. And well, thank you very much for organizing this.
Thank you, Eduardo, for sharing with us your expectations. We would like to know if there are also like students connected in this tutorial. What do you think, what you are expecting from this tutorial? Don't worry, we'll be here to hear from you. So if anytime if you have something to share with us, don't be shy. We will be here for hearing. So let me start with the first part of today is what is called the design and synthesis of nanoparticles for nanomedicine. So I will give like a brief introduction related to nanomedicine. I think that is something that you already know, but it's coming from nanotechnology. What is nanotechnology? If we look to the origin of the word, it comes from Greek. This nano prefix means drop, or something very, very small that is like 10 to the minus 9 meters, something that we cannot see without the help of powerful microscopes. And nanoscience is the study of these structures that are this tiny size or molecules. Let's say that from ISO is the size is ranking between 1 to 100 nanometers. And the application of this science is what we know as nanotechnology, what we pass through the real application of the knowledge that we are creating from nanoscience. There are a lot of applications, so we can find nanoscience and nanotechnology in a lot of sectors. For instance, we are interested in biomedicine, but it's not the only application that we can find from nanotechnology. And with this map, what I want to show is how important is the interdisciplinarity in nanotechnology, nanomedicine. So we have to work with different disciplines to understand mechanisms and the application of nanotechnology. Briefly, let's say that the circles that are bigger, let's say, are more studied, and then we have connections to develop this kind of nanomaterials. So for instance, we see that the major contribution in these categories for nanomedicine is biomedical science, chemistry, material science, and environmental science. So the home message is like collaboration. I think that is the only way to reach nice results in the benefit of society. So it's a key point in nanotechnology, nanomedicine. So as brief introduction to now really nanomedicine, that is the use of these nanotechnological tools to diagnose, prevent, or treat diseases. I would like to share with you a short video of for nanoparticles that are actually used in clinical trials. So don't forget that the ultimate goal of nanomedicine is really to improve the quality of life of patients, of humanity. So I will just share with you this video. Please let me know if you have the image and the sound from the video.
Imagine something 80,000 times smaller.
Could you hear the video?
Yes. Breath of a on a fingertip, unlocking a new frontier into cancer research. Nanotechnology, the science of building small, is changing the way we look at cancer. More importantly, the way we look at diagnosis and treatment. Nanotechnology allows researchers to build new tools that are actually smaller than cells, giving them the opportunity to attack cancer at the cellular and genetic level. This technology not only enables health practitioners to detect cancer earlier, but also holds the promise of stopping cancer before it even develops. This revolutionary approach is so precise, doctors will be able to design a unique treatment for an individual's own medical and genetic profile. Based on computer chip technology, diagnostic devices such as nanoarrays are thousands of times more sensitive and accurate than current techniques. Because of their size, multiple lab tests can be done more rapidly and at a much lower cost, using one nano device instead of many. Nanoshells can be linked to antibodies that recognize tumor cells. Once they are taken up by the cancer cells, near-infrared light is applied, killing only the tumor and leaving neighboring healthy cells intact. Scientists are engineering nanoparticles, such as dendrimers, to seek out and destroy cancer cells. This amazing technology can be customized for targeted drug delivery, improved imaging, and near real-time confirmation of cancer cell death. Moving research from bench to bedside is an important goal of the National Cancer Institute's Alliance for Nanotechnology in Cancer. A collaborative plan is underway to share research and development information across scientific disciplines and around the world. As biomedical applications of nanotechnology evolve, scientists are ensuring that nano devices are safe for both the body and the environment. The National Cancer Institute is optimistic that through coordinated and responsible development, nanotechnology will dramatically change cancer patient care. The science is at our fingertips.
So I think that this video summarized which is the purpose of nanomedicine, the diagnosis, prevention, and treatment of diseases. So I hope you enjoy, and we will be speaking about the principles of all these nanomedical tools during the tutorial. So if you are ready, and if you are a student, maybe you will be happy to see how are your knowledge in nanomedicine. So I invite you to scan this code and to answer a questionnaire to have a starting point in nanomedicine. You can do it now, you can take it for later. So there are general questions about nanomedicine, the application, the use of nanoparticles. And to go through the part of synthesis, so we will find different approaches to develop these nanoparticles. And something that we were watching in the video is the more powerful approach of theragnostic. So in one single nanoparticle, nanoagent, we can combine different properties. For instance, the diagnosis and the treatment of a disease. Particularly, we have been seeing the last years that cancer nanomedicine is the most studied application of nanotechnology in this field. So we have different modalities, different possibilities of using nanoparticles for medicine. For example, or the main goal is to overcome natural biological barriers or limitation, physical biological limitation of conventional therapies. So for instance, when we are talking about system that is used to deliver a drug, so what we want to overcome is the no water solubility of some drugs or the lack of a specificity of the drug, and then we have to design and synthesize nanoparticles to be used as vectors that we can really deliver locally for an enhanced performance of the therapy. So let's go to the synthesis possibilities approaches. So generally speaking, this is an overview of what we can find like in nanoscience, nanotechnology. So we have two routes. The top-down route that we are taking like a bulk material, and then we break down this in smaller parts of the bulk, and then go to the nano scale. Or we have the bottom-up approach that we are taking like units, small units, molecules, atoms. We start with these entities, and then we start to grow in a cluster. Some atoms, molecules comes together, and then we have finally like a nanostructured materials. So there are the two main approaches used to synthesize nanoscale structures that have different advantages and disadvantages. For instance, we have for the top-down approach that, okay, we can use these techniques to develop or to synthesize in a large scale these nanostructured materials. And because we are using like pure bulk materials, so there are not necessity to use techniques for the chemical purification. Disadvantages of these kind of approaches are that we have a broadside distribution, so we can't control precisely the size and the shape, the geometry of the final nanotechnological product. It's difficult to control parameters to achieve like this homogeneous size and shape of the nanomaterials, and we can have some impurities related to the geometry. Finally, disadvantage. So most of these techniques are more expensive than, for example, the second approach that we have, that is the bottom up. So one advantage is that we can control more precisely the size, the shape. We can produce different kind of nanomaterials. We have narrow size distribution, also like soft 20 nanometers nanoparticles. These techniques are cheaper than the previous one. Disadvantages of using bottom up is sometimes we cannot have large scale production of these materials through the bottom up technology, and we will need chemical purification. We are using some substances that will help us to define the oxidative state of the nanomaterials. So we will need this multiple stage production. Through these approaches, so we can have different nanomaterials that can be classified based on the dimensionality that they have. So if it's different dimensions, for example, we have particles, nanospheres, or different shapes like tubes, rods, and films. So we can use these techniques to develop different dimensions in the nanomaterials. We are going now through some of the techniques that are used to synthesize the nanoparticles, and there are dividing depending of what we are using, if we have just physical changes, chemical, or what is happening during the production. So mainly physical approaches are classified like a top-down strategies. So we have, just to mention some of them, there are more. You can take a look in literature and see which are like the necessities to develop these techniques, which kind of materials we can develop with them. So we have laser abrasion, high energy milling, inert concentration, pulse vapor deposition, flash spray pyrolysis, electro spraying. So we are going just to see some of them. For instance, what we have in laser abrasion is like, in this method, we will use ultra short and high intensity focused laser beams to destroy, let's say, materials from the bulk that is target that may be immersed in a solution in a aqueous medium to form a nanoscale nanomaterials. So here is just a short video that shows how we can form nanoparticles from the bulk with laser abrasion. We can produce, for example, gold, silver, or different metal-based nanoparticles from this technique. Something important is that we can adjust the characteristics of the technique to make it greener than other processes. This is just the principle of how this process works. That, as I said, we have like a target metal, a bulk of metal target that is irradiated with a laser, and then we have different spatial phenomena occurring close to the target. We will have the formation of bubbles that will explode, and then we have the production of the nanoparticles with these chemical qualities of the target. So no further purification is needed. Then we have, for example, high energy milling. So it's like we have powders that we will mix, and that they will be subject to a high energy collisions between balls of different materials. And then because of this mechanical energy that is put in to the powder, we have like chemical reactions that will occur in situ in the powders. So it's a very interesting technique also, because we can develop greener technologies with this kind of approaches. We have uniform dispersion, for instance, of oxide nanoparticles. Then we have some of the techniques that are used for a chemical production of nanomaterials, taking into account the bottom-up technology or approaches that are used. Just to mention some of them: co-precipitation, chemical reduction, for instance, of metal leak salts, electrolysis, emulsions, microemulsions techniques that are a lot very interesting also for nanomedicine. In fact, photon radiation chemistry, xenochemistry, soil gel, and other techniques as hydro and soil thermal synthesis. Just to say some general principles of the techniques, we have photon and radiation chemistry. So these techniques are using different kind of photons or accelerated charged particles that may be ionizing radiation, like UV light, electron beams, X-rays, gamma rays, so it's carbon, for example, ions. And when we excitate or we irradiate a solution with this kind of radiation, we create an excitation and ionization state of the water molecule that will produce oxidative or reductive radicals, so that are very strong reactive species to develop, to synthesize nanomaterials. So in principle, for example, we want to create metallic nanoparticles by photoradiation chemistry. So the main species that are involved in the reduction of the metallic salts are the solvated electron and the hydrogen radical. So these processes happens in a very specific time and space scale. So there are ultra fast processes that allows controlling very nicely the size, the shape of nanomaterials. For instance, I'm just illustrating here the formation of gold nanoparticles that comes from ions that are irradiated in solution. And after irradiation with the qualities that I just mentioned, we can obtain metallic cereballen, let's say, gold nanoparticles with different shapes, depending on what we are introducing in our system. That can be just water to make a greener strategy of photo or radiation chemistry, or a co-solvent to avoid some processes that may reduce the yield of the reaction. So it's a way to produce metallic nanoparticles and with greener strategies. Then we have, for example, sonochemistry. Also, I choose this technique to introduce to you, as we can use it like a sustainable alternative of the production of nanomaterials for nanomedicine. In this case, the fundament is the use of high intensity ultrasound. So maybe there are some techniques that you are using in your lab, so you are learning if you are a student. So this high intensity ultrasound can be used to produce these materials that can be in situ coated with some interesting molecules. So it's a route that will not need like high temperature, pressure, or long reaction times like the conventional chemical approaches. As in a specific or well-localized space, we can create different phenomena by interacting this ultrasound with the medium and produce the nanomaterials. So generally speaking, what we will have is acoustic cavitation. So we will have like a first step of the sonochemistry and a second step when we are producing like bubbles with a high energy, Talmar energy, that will catalyze the chemical reactions. In this cartoon is just like a representation of what we have for the ultrasonic irradiation approach. It's the same idea. We have precursors with ions, metallic ions in this example of gold 3+. And after irradiation of the medium, we will induce the formation of all these stages in solution that will give origin to the growth of the nanoparticles. I will just invite you, if you have time, to visit this poster, P033, that talk about the development of gold nanoparticles with quercetin. That is a natural extract, and how the toxicity was evaluating for some cancer applications. Again, is on the way to develop and use more, greener approaches of nanoparticle synthesis. Another approach that we have that is commonly used to synthesize not cereballen metallic nanoparticles, but metallic oxide nanoparticles, is the sol gel technology. That is like a wet chemical technique that follows a well-established steps of production of reaction using some specific precursors of the metals that are called metal alkoxides or metal chlorides. So with this technique, we can, for instance, synthesize silica nanoparticles, titania, zirconia, all these kind of ceramics that are also very interesting for different applications, not only nanomedicine. So the steps that are followed the reaction is hydrolysis, polycondensation, gelation, edging, drying, densification, and crystallization. So we have the combination of some molecules at the beginning in a solution that goes the formation of a solid state. The main advantage of this sol gel technique is that we can adapt this technology. We can change some parameters such as pH, temperature, precursors, solvents, quantities or ratios between aqueous and organic phases to induce the formation of different textures. Let's say, for instance, non-porous metal oxide nanoparticles, mesoporous that are a classification of some textures with porous ranking in a certain scale. Then we can produce, for instance, hollow mesoporous silicon nanoparticles if we are using a precursor from silicon, core shell mesoporous particles, George shell or Hancus mesoporous particles. And all of these structures are really interesting in developing, for instance, some vectors for drug delivery. So this is like the main advantage of this technology, the sol gel, that we can modulate the texture of the materials. However, it's not as greener than the previous possibilities, using some stimulation with radiation, ionizing, not ionizing radiation. So this is the main disadvantage, and also there is a multi-step synthesis. So we have to use a lot of time and resources to develop this kind and these nice structures. So we have to balance what we want, depending of the design and the application that we are projected for medicine. So finally, another approach is the biological synthesis that is also classified like a bottom-up mechanism of nanoparticles synthesis. So we can use, for example, microorganisms to generate these materials, or active substances from different plant tissue. So there are the main two approaches that we can use. And by coming from natural origin, or by coming from natural extract or microorganism, so it's an eco-friendly approach to the synthesis of nanoparticles. Most of the time, this approach is used to synthesize, again, cerevalent metallic nanoparticles, as some of these substances that comes from natural origin have this potential to reduce metallic ions, and to coat the surface of the nanoparticles. So it's very interesting option also that can be developed in one step or multiple steps, depending of what we are looking for the application. Now that we know, we have like a general idea of the development of nanoparticles, so we have also a way to classify these nanomaterials for medical applications. So generally speaking, we will have like the group of the organic nanoparticles, the group of the inorganic nanoparticles, and in between when we are combining properties from an inorganic nanoparticle with organic coating or organic with inorganic, depending, we have what we know like as hybrids or of these nanomaterials. So I'm just showing some of this classification, organic, inorganic, and particularly those that are actually used in clinics for treatment of different diseases. In the case of organic nanoparticles, the most common and the first kind of nanoparticles that were used for this kind of applications were the liposomes. And for instance, in organic nanoparticles, so we can find in clinics gold nanoparticles, iron oxide nanoparticles, and hafnium nanoparticles for this kind of materials with this chemical or this element, hafnium, is particularly used to enhance the performance of radiation to treat solid tumors, well-localized tumors. So just to give a brief overview of what liposomes are and why are they interesting for nanomedicine. So they are nanoparticles that consist of one or more lipid bilayers. The main advantage of having these layers is that we have a space to be used for low water-soluble molecules or drugs, and we will have like a domain that will allow us to load or encapsulate molecules that are not soluble in water. So this is with the idea of overcoming this limitation of most of the conventional drugs that are used actually in the treatment of different diseases. So the synthesis of this kind of nanoparticles are, we can follow different methodologies. Here I'm just illustrating one of them. So in short, it's like to have the lipids and, for instance, a basin or the drug in organic solvent. Then we have to remove the solvent. We will have like dry lipids that will follow a hydration process, and then dispersion, like to from one conformation go to simpler liposomes that will be able to carry, in this case, the basin for a much more precise treatment of infection, for instance. Here I'm just presenting like the general classification of this kind of nanoparticles. So depending of the conformation of these lipidic bilayers, so we have like a small unilamellar vesicles or liposomes. We are in a size range from 20 nanometers, for instance, to some micrometers. Then we have large unilaminar. Then if we have several layers of these bilayer liposomes, so we have a multilamellar and different conformations. And based on the composition, we can classify liposomes as conventional or photogenic liposomes, pH sensitive, cationic, or some of them can be, let's say, coated with other molecules, macromolecules, for long circulating developments and for immunotherapy. There are some examples of the use of liposomes in the treatment of different diseases, or in this case for medical applications. So the flexibility of organic nanoparticles is based on of its surface that can be conjugated with different species to give some specific task to the liposome for the delivery of drugs. So it's just what we want to represent in this cartoon. Like, we have a lot of possibilities. We can work in the surface to give some enriched properties to the liposome. And here I'm presenting some examples. For instance, the use of liposomes to encapsulate antineoplastic drugs used for cancer treatment, very conventional and widely used in clinics. Doxorubicin, vincristine, using for different kind of cancer. Or for instance, to encapsulate iron oxide nanoparticles to be used as contrast ions or for targeting therapy. And why not to mention that liposomes are also participating in the development of the COVID-19 vaccine. So Pfizer and Moderna use this kind of nanostructures, organic nanomaterials, for delivering what will have to fight against this pandemic. So they are widely used, as I said, for the delivery of different substances, and thanks to the surface, we can give other properties to improve the circulation of the liposomes. Regarding inorganic nanoparticles, we have metal-based nanoparticles. Why I'm presenting like this kind of nanoparticles, or why I'm emphasizing in this kind, I think that you will have the opportunity during this Congress to see all the research in this area, as metal-based nanoparticles have very interesting properties, optical properties, to be used in medicine, like for the treatment and diagnosis, or like in a biosensor for detection of different microorganisms. So that's why it's really interesting, the use of, or to talk about metal-based nanoparticles. And in the case of the chemical reduction of these chemical reduction process to prepare the nanoparticles, so we will require, or it's necessary to use a chemical reductor, let's say, as we have to go from metal ion to cerebralin metallic nanoparticles. So here I'm just showing some of the reducing agents that are used for create these metallic nanoparticles, and depending of the nature of the element of the metal, we can find, like in the literature, which are the most common reducing agents that are used, if there is necessary, of which kind of stabilizer agent can be used to control the size and the shape of the nanoparticles. And in this summary table is like, if we are following this synthesis with these precursors and reducing agents, we have different characteristics. For instance, the size of the final nanoparticle. So we have like the most common and widely used processes to synthesize the nanoparticles. I'm just showing here how the Turkish method that is very famous, that you citrate reduction of gold salt is used to produce the nanoparticles. In this case, it's important to mention that sometimes there are like some competition with at different moments of the formation of the nanoparticles, when they are in the nucleation step, in the growing step, and then for the final formation of the nanoparticles. So it's important to take into account which precursor we're using, the ratio with the stabilizing and reducing agent that we are using to obtain homogeneous colloids containing the nanoparticles. So is the Turkish synthesis going? And then more sophisticated methodologies following the chemical reduction of the metal salt is the use of other macromolecules, let's say polymers, that particularly have a tile motif that will have a direct interaction with the surface of the metal to make it more stable and to have a covalent coating of the nanomaterial. Or for instance, if we have this first step of the coating, then we can conjugate with different molecules to give a specificity to the vector, to the nanoagent for a particular application. So just to summarize a little bit and to have an overview of the importance of functionalization, what it means that how we work in the surface of the nanoparticle to amplify the possibilities of the nanomaterials. So is that we have to take a look first in the size, the shape, some physical properties like mechanical properties or textural properties, then how our nanoparticles is composed, for instance, if it's organic, inorganic, hybrid nanomaterial. Then in the surface, what we are using to reduce toxicity, to give a specificity, so we can start with a very simple coating and then working on the chemical functionalization. So using different groups, we can conjugate with a specific ligands or antibodies, polymers, or for instance, peptides, aptamers that will be recognized by tissue, by cells, and to be more or to enhance the delivery of the nanomaterial. And then we have, like, if we are working in the design of these kind of nanoagents, we have what we know as smart nanoparticles, that this concept is coming from how we can deliver or release the active substance that we, for instance, can encapsulate in a liposome. So we work in the surface, and then maybe some of these molecules can be stimulated or by external beam and to degrade and deliver the substance. So these smart nanoparticles can come from some biochemical or internal trigger, let's say, biochemical reactions or changes in the pH, or as I just mentioned, external stimulation using maybe light, ultrasound, a magnetic field, changes in the temperature, and then we have like a very smart particle that will allow us to increase or enhance the performance of a conventional modality to treat cancers, for instance. And why is so important this part of functionalization? Because depending on what we have, that is coming from different steps in our synthesis, or can be developed in a single one-pot step, we don't know. It's like the real goal of designing these materials. So we can have like passive targeting or active targeting of damaged cells, of unhealthy cells that will interact with the tissue of the tumor cells differently. So if we are talking about passive targeting, so you will see later in more detail with Dr. Juan Vivero. So we can like enhance the circularity, how it's coming from the body to the target side, or active targeting. I just just for an effect that allows this circularity, or active targeting, what we are really including a particular receptor that will identify the unhealthy cells, and then in the place, or place it in the tumor or in the malignant size of, we can now stimulate and deliver something. So that's why it's very important to work with the functionalization and go through different steps to validate the use of a nanoparticle for a treatment. So in this part, we are like having a summary of some of the main methods to synthesize, and then when we have a nanoparticle, how to improve the properties with the targeting, with different actions that can take. And now let's talk just a little bit about this concept that is used, safer by design, that I think that is a really nice alternative that we are, for instance, me, young researcher, or students, or as you researchers in different centers in all around the world. So we have to think about is like not only to use a technique by using a technique that will give us some result. It's really to think about everything from the beginning by designing the particles or the tools that we want to use for helping humanity, in this case to enhance the quality of life of patients that have, for instance, cancer. So it's important to see all the risk that is involved in the development, in the production of the materials. Think about the whole life cycle of the nanomaterial, identify what we can improve, how are the different strategies. So it's to see the life of the nanomaterial from all the point of views, to have like really engineers in greener engineers in this area. And in order to have this safer by designing the nanoparticles or the nanotools used for medicine, it's important to think about green chemistry, as I was mentioning, different approaches. So for instance, green chemistry is indeed the design of chemical products and processes that will reduce or really eliminate the use or generation of hazardous substances. So that's the key point of green chemistry, and that will be applied in the life cycle. So it's completely connected with this new philosophy of safer by design and green chemistry. It is now also a sustainable chemistry. These are just to remind you the 12 principles of green chemistry: waste prevention, atom economy, less hazardous chemical synthesis. So we can apply this really not only to organic chemistry synthesis, but also to nanomaterials for medical applications. So there are some key points that are coming from green chemistry. So it prevents the pollution. It's really a philosophy. If we are start in our labs with our students, like to encourage to adapt the techniques that we have to be more sustainable. So it's really important at this point. And why not, like by applying these new philosophies, we are really contributing to the sustainable development goals. in the benefit of humanity. So this is the first part of this tutorial about an overview of nanoparticle synthesis. I would like to stop a little bit here to hear about you, your ideas, your comments, how you are applying in your labs, maybe green chemistry, which are the techniques that you are familiar with to develop your materials for these applications. So if someone wants to participate, will be really nice to hear from you.
Thank you very much, Daniela. I just wanted to, well, to mention that it was a very nice introduction to nanomedicine. Well, in my case, in my lab, we have just started with this green chemistry and all the principles that the universities are forcing researchers to take care of. It's just the very beginning, so no use of solvents, just save water, save energy as much as we can. And then dealing with the suppliers to see which is the most sustainable raw materials that we can use. So it's the very, very beginning. But the point that I wanted to mention is that I found it very interesting that you brought up the liposomes as an example of these nanocarriers for vaccines. In the case of COVID-19, for example, by talking to some colleagues recently, on one side, I had the feeling that this new development of liposomes to suddenly allow the delivery of messenger RNA for this new technology in vaccines would be a boost for nanomedicine. But then on the same side, I'm also hearing from other colleagues that is also bringing the attention of the risks that nanomedicine can bring into the products as well. Like on one side, we hear about liposomes being generally biodegradable and being very useful for the delivery of these molecules. And on the other side, we also hear about nanoparticles of graphene oxide that could be very toxic and could be included in some of the products. So I still have the doubt that the public is accepting generally nanomedicine, and it's more and more, let's say, commonly welcome in society. Or on the other side, there is growing this doubt generally that what we are doing in the lab is still a bit hidden and a bit mysterious. So I just wanted to mention that point.
Yeah, thank you, Eduardo. Yes, I think that in Mexico also we have like to explain to the population how is the nanotechnology and nanomedicine working, and now more because of the vaccines that they are already nanotechnology there, and it's important to educate, I don't know if it's the word, the society with the use of nanotechnology. We will have the yin and the yang. Yes, no. Thank you. So let's continue with the next part of the characterization, if there is not any other comment. So I prepare also like a small, some questions about characterization. If there is any student, it's like a nice starting point, like how much do I know about this chemical characterization techniques. So it's again like an overview of some general principles of the techniques. So to start is like to see that when we are developing nanoparticles for medicine, so we have to pay attention in different aspects, starting with properties, the physical-chemical characteristic that the nanoparticle or the nanomaterial has, and then what's happening regarding the pharmacology, toxicology, and immunology of these materials. So we can use, for instance, different models that we will see later with Dr. Juan Vivero. So in the case of physicochemical characterization, so it's important to identify different characteristics, the size, the shape, the chemical composition of the material, the molecular weight of molecules that we are using to grow these materials, the surface chemistry that, as I mentioned, is a key parameter for the surface functionalization and the targeting of the materials. The purity, stability, solubility, there are maybe some other properties, but it's like the main that we have to know. And then you will see how important are the in vitro and in vivo characterization to have this path to the clinic. So generally speaking, we will have like the characterization of the molecules that we are using, then the nanomaterial that we are producing, and in between we have this really interesting physicochemical characterization. In the case of the molecules, so we can start by analyzing the chemical composition by different spectroscopy techniques that complement the information about the nature, the chemical nature of the molecules, the materials. You may recognize some of these acronyms, like coming from the techniques that are used. Then we have to take a look to the nanomaterials. So we have microscopy techniques that can be electronic electron microscopies. We have different spectroscopy technique, like to see really the size when it's in solution, for instance, or to characterize the surface charge or some optical properties of the materials. So it's interesting that when we are developing these nanomaterials, so think about what we want to know, so depending of the final application. So what to measure is like something really important to think about, as we will define or find the way to apply this nano tool by measuring different things. So size, shape, surface charge, and for instance, the texture or porosity of the different materials that we are developing. So I normally like to think about the electromagnetic spectrum as the main thing to choose, for instance, a technique to characterize or to elucidate the properties of the nanomaterials. Why? Because most of the techniques that we are using, they have like an input that is energy to create different phenomena, or that would allow us to know more about the chemistry of the matter, generally speaking. So depending of the energy of these photons that are used, these electromagnetic waves, so we can know more about the molecules and the nanoparticles. If we are using, for instance, infrared radiation, so we will know more about the surface, the functionalization of the nanoparticles. If we are developing some nano tool and we want to add a specific functional group, so we can use this as, by nature, infrared radiation interacting with matter will allow us to know which is binding to which, and to know more about the chemistry. Then if we want to know about different electron excitation and other processes coming there, so we use visible and ultraviolet basic techniques, let's say, and then ionizing radiation to know more about the electronic state of the material, or to create some images coming from different interaction of matter with ionizing radiation. It's just to have an idea. So for this part, I was based in this article that I found really interesting, because it's summarizing most of the techniques that we are familiar with in this area to characterize the nanoparticles. So they are presenting the different aspects that we have to know in general for material science. So if we are talking about size and size distribution, so we can use different techniques that could be microscopy, spectroscopy, or other theoretical models that are really complementary techniques to elucidate these properties. For instance, we have a sphere nanoparticle will be different, or we just need one parameter to define the size of this morphology. And for different geometries of nanoparticles, so we have to know more aspects to play a little bit with techniques to know all the axes and dimensions of this. Is something that is called also aspect ratio. So to know if we comes from a sphere to another geometry. And also, for example, to see the real size, so we have to think about the method that we have to use to determine this. So it depends what we are looking for, that we can just use the radius of gyration that we can obtain by using some microscopy techniques, or the hydrodynamic radius that is more interesting when we are talking about nanomaterials developed for medical applications, as most of the time, as we already said, we are using ligands in the surface to give specificity to the tools, to the vectors. So we have to know really the size of the complete tool. And then to see the size distribution, so depending also in the technique that we are using. So we will have from the mean size of the particles, different distributions. And depending, for instance, if we are thinking of in the number or volume of the particles, we have different distributions. You can take a look to this kind of information when we are using, for instance, dynamic like scattering. So it's different ways to identify, to characterize the size and size distribution of the nanomaterials. And for the shape, I think that it's something common as a human being. Like if I want to know the shape, I can see it or I can touch it. So in the case of nanomaterials, so we have to see. So nanomaterials, for this characterization, we are using what is transmission or scanning electron microscopy. As by the size of the nanomaterial, we cannot use optical microscopy from the size of the wave. So here just showing the principles of using electron beams to have an imaging. So depending of the interaction at what we are collecting from this interaction from the primary electron with the target, with the surface, we can obtain images if we are collecting, for instance, secondary electrons, or if we are producing other subatomic particles. So depending on what we are collecting is the information. Very interesting that when we are using electron microscopy is that also we can have an idea of the chemistry. So if we are exciting electrons from the inner shells of the atoms, so we'll have different information, X-rays that are emitted, and we can collect and know more about the chemistry. So these are just the fundamentals of the techniques. It's also, for instance, when we are using for the surface, depending of the how the radiation is penetrating in depth, so we will have also information of the texture or the of the material. This is again like how this technique works and the information that we can have if it's scattering, scanning the surface, of if it's transmitted, and we detect later the information to form an image. So if we are using transmission electron microscopy, we will have like very nice resolution, and we will know about the morphology, the shape of the material. But it's important to take in mind that it's just a 2D projection of what we have. So depending of the orientation of the beam, we will have information in 2D. But if we are like combining with the scaffolding based techniques, so we can have really like more information about the shape of the particles. Another complementary technique based on microscopy is atomic force microscopy. So I'm just illustrating the principle that we are using a tip that will be going through a surface, and we can have atomic resolution. So depending of the forces that act in this tip that will touch the surface, we will know more about the mechanical properties and also the shape of the materials that we can characterize by this technique. And there are different modes. I invite you to take a look to the technique if you are interested in. Then if we have already like the size, the shape or morphology of the particles, so another interesting point is to know the surface charge of the nanomaterials that we are developing. So for this, we can use like zeta potential technique that based in how ions or ligands are attached to the surface of a nanoparticle. We can determine by the mobility of these ions, the surface charge. We can also characterize the texture of the nanomaterial, so the porosity, using techniques that takes into account how a gas is absorbed in a surface, how this gas fulfill the porosity, and then it's just like the way to know how is the texture of the nanoparticle. We can obtain information about the size, the volume of this porosity, and there are really interesting these materials for drug delivery, for instance, because we can modulate or chip the size, the shape of the internal and external domains of porous nanomaterial. Something very important also in the part of characterization of nanomaterials characterization is how we prepare the samples. I think that is the challenging point in this part of knowing the properties of the nanoparticles. It's, in my experience, something really important, as we can do different or to prepare different samples, and sometimes it's not what we want. That's why I also saying like it's really important to know that all the techniques are complementary and how to prepare this. I would just play the different videos that I choose to show. Is, for instance, for the two main modalities of the electron microscopy. So in this case is for scanning electron microscopy, what we are looking for a surface or materials that we obtain as powders, or in the other side we have like nanoparticles as colloids. For instance, gold nanoparticles that are electron dense materials, so we can really see by this technique. I think that there are different ways to prepare, just with a drop or difference. I can share with you the different videos that I'm showing. But I think that we can continue. It's just this table, a summary of the techniques that we can use to characterize the nanoparticles when we are using dry samples. So particularly is when we are using ionizing radiation, electron beams, X-rays, that we have to focus the beam in the sample. So it's important to know how to prepare the samples in dry conditions to evaluate different properties, size, shape, crystallinity, or also chemical properties or characteristics of our nanomaterial. We have some of the techniques that are used in suspension. So when we have colloids, we can take a look to the size of these, the dispersity, the charge, as I just mentioned. So this table is showing the summary of the techniques that we can use. And why it's very important to know these properties or characteristics of the nanoparticles is because depending of what we are using, the nanotube that we are developing, the size, the shape, the surface charge, the texture, is how at cellular level they will be internalized by the targeting cell. So we have different pathways to uptake pathways, let's say. So depending of this characteristic is what we are expecting, where are going once inside the cells, and which are the biological consequences of the nanoparticles. In this cartoon is the same. All these characteristics are very important, but now, and from the point of view of more complex biological systems, so not only for the uptake in cells, so really the distribution once they are, for instance, injected to a patient or to an in vivo model. So it's important to know these characteristics. If the cell have here negative charge in the membrane, so the charge is important to enhance the interaction. How is the circulation of the of the nanoparticles inside the body, how we can eliminate these nanoparticles. It's relevant information that we have to take to evaluate for future applications. So in this part, it's just some agencies and some links that you can also take a look where there are more information about like well-defined ways to characterize nanomaterials for nanomedicine. That's all for this first part, synthesis and characterization of nanoparticles for nanomedicine. Was really brief, the last part. I will give you the two documents to help you in the characterization part, like to have a guide if you are interested, if you are doing the same in your labs, and how to improve the way of characterizing your potential nanotherapeutics. So I will stop my screen, and I don't know if there is any comment, question, or something from your part.
Hi, my name is Daniel Garcia.
Hi, Daniel.
I'm from Universidad Autónoma de Coahuila. I'm working with iron nanoparticles as support for protein extraction. But I have a doubt. What are the considerations to take into account when we want to develop or synthesize our nanoparticle from microorganisms or plant extracts?
Thank you, Daniel. Considerations do you mean by synthesis conditions that you can work with in order to obtain the nanoparticle?
Yes, and how can I select specific microorganisms or a specific extract plant?
Okay, so regarding the conditions, you can play with the ratio between the extract and your precursor. In this case, your iron precursor, pH, temperature, reaction time, all these that are coming also from chemical synthesis of nanomaterials are conditions that you can modulate to obtain the nanoparticle that you are looking for. Okay, regarding the source, the natural source, the bioactive substance that you will use. So it depends, and I would invite you to take a look in the literature which are the best. They are used to reduce, to stabilize, but in the case of binding a certain or a specific protein that is your application, so you need to find the other part that you will need. So it will depend of the application, which synthesis or which bioactive substance you will need from the plant, for instance, or which microorganism you will need.
Okay, thank you.
You're welcome. I don't know, Dr. Juan Vivero, if you have another comment to this question. So.
Thanks. No, well, I was thinking, I guess, thinking more. I guess what you understand from your question is in terms of microorganisms. If you were targeting specific microorganisms, what we have done in my group and we have found very helpful is target those nanoparticles or even single molecules with a positive charge. That similar to mammalian cells in terms of microorganisms. And here I'm talking more in bacteria, some type of bacteria. It has been very helpful in terms I'm saying targeting, just with quotation marks there, because of course that target, or that's not that specific. Of course, there are other ways to make a target specific, but anyway, that's a general approach, just putting positive charge in your nanoparticles. But I'm agree with what Dr. Daniela said about the different strategies in terms of natural products, and also thinking ahead of what is the final application of these nanoparticles. Thanks.
Thank you. Yes, because sorry, we in our group we try to synthesize some nanoparticles, but we use microorganisms, fungus, that is Fusarium oxysporum. But the main challenge of the synthesis is to recover the particles, because it seems that the Fusarium produce inside of its cell of itself. So, but without kill the microorganism to produce more. So that is the reason that I asked that if you have some experience or some experience synthesizing some particular like this, or using another microorganisms, but if the challenge that we have, you already have.
Jump quickly here, just to, I guess I misunderstood the question. Sorry about that. I thought you were targeting microorganisms, but you are using microorganisms to make nanoparticles. My group doesn't have much experience on that approach. You say that, I believe you say these are magnetic nanoparticles, right? Iron oxide, I believe that you mentioned that. Just as a comment, again, without having too much experience on that, well, if they are magnetic nanoparticles, you can take advantage of the magnetic properties, of course, to pull out the nanoparticles from, and pull out, I say you have to, and again, coming from somebody who doesn't have much experience there, but maybe I can imagine you can utilize the microorganisms and pull out the nanoparticles, and that might help you to improve the recovery of those nanoparticles. But that's just a general comment.
Sorry. Thank you, doctor.
Thank you, Daniel. So now we continue with the next part of biological characterization. So it's 11:08. If you agree, we can take now a coffee break, 15 minutes, and we meet together soon to continue with the biological characterization of nanomaterials for nanomedicine.
Thank you. See you in a minute.
Thank you, Daniel.
Hi, Dr. Daniela. We are going to continue, or we're going to do the recess?
We have 15 minutes coffee break.
in some kind of biological medium. So that's what is really important to take account of those characterizations. And just bring it back, this slide that Dr. Daniela already presented in a few minutes ago, how those physicochemical characterization can be related to some outcomes in vivo or in these biological applications. And these data or these trends, because at the end some trends, we cannot speak for very specific, I guess, numbers, or I mean, like, as you can see at the bottom of the slide, there are some sizes there that address different behaviors in the biological application or the biological performance. In this case is, for instance, EPR. I'm looking at the EPR effect, accumulation of those nanoparticles in tumors, talk about 220 nanometers or so, and those numbers has changed. In my understanding, people now is talking more about 50 nanometers hydrodynamic diameter of the nanoparticles. That's kind of the optimized or best size for targeting EPR. Anyway, these are some trends, and those are useful. And those, and the main point is those values or those characterization from the nanoparticles can give you some particular idea of how they behave in the application. Again, it's mainly therapeutic application, and in humans. I guess that will be the ultimate outcome. That I should disclose that right now, most of the nanoparticles that I'm going to present, or not the nanoparticles, sorry, the slides that I'm going to present are related to cancer treatment. That's kind of the criteria that those nanoparticles follow, but the concepts are pretty general and you can apply to other fields. Okay. So in vitro, and here in vitro, the term what I'm using is cell level. I know that other fields might use in vitro, in vivo, in other concept or in other terms. For me, in vitro means experiments or characterization at the cell level. And in vivo, the term that I use in vivo is for animal level or animal experiments. So there is a huge cascade of characterization techniques that you can do to evaluate your nanoparticles using for in vitro characterization, and those cascades can keep growing depending of, I guess, of your nanoparticles. There are very specific nanoparticles that some of those features are more important than others. The ones that I'm gonna describe more in detail are gonna be, again, more related with cancer treatment or so, but I just wanna point out some of the, for instance, some of the techniques that look at the macrophages function. All these in immunotherapy, immune response of cells are really important. I have a collaborator that works with siRNA nanoparticles that one of the main issues with siRNA, as far as I understand from his research, is the immune response that has seen in patients due to these technologies. So he's really focused on testing his nanoparticles or the immunological response of those nanoparticles. That can be tested in vitro level. There are specific cells that are used for that to generate or those cells be stimulated to generate cytokines, IL-6, IL-8, other type of immune response, and he's looking in depth to that. Others are more specific about necrosis, oxidative stress, because those nanoparticles that are developing are more focused on that. So again, I don't want to take you to take this at least as the ultimate list of all the in vitro characterizations that are out there, because again, those which I try to put together most of those techniques that people do, but there are some others might not be here, or there are some of these techniques that people might not even never do this characterization, because that is not might not give worthy data for their platform. So just focusing on the ones that we use more, again, for cancer treatment, and maybe Daniela and Eduardo might have some comments there that they might use different ones or that find very important. But the main ones that we use, at least in my lab, is the cytotoxicity, or testing the cytotoxicity of these nanoparticles. The most common that we use is the so-called MTS or MTT-assay that looks at the metabolism of the cells using these specific salts inside the cells that, again, look at the activity of the mitochondria inside the cells. And what it does, or what we do, is based on control experiments, which is cells without treatment, it will give us a specific amount of metabolism. And this metabolism is, again, these salts actually, they absorb a very specific wavelength in the visible that is quantified with plate reader. And so we compare that with cells that hasn't been treated, as we call those 100%, and the cells that has been treated, well, in principle, should be less cells, so the metabolism of those cells, the value of the metabolism of those cells should be reduced, and that's compared with that 100% of the control, should give us a reduced data in terms of the metabolism. Again, all when we report that in papers, we call it that percent viability. Sometimes we get confused in the field. We think percent viability is actually kill cells, which most of the time is kill cells, but this technique per se doesn't look at dead cells. Again, it looks at metabolism, and it relates higher metabolism with higher amount of cells, lower metabolism with lower amount of cells, and again, relate that with viability. But that's very common technique, and again, we do all the time. The other technique that we can use is using the Guava Test. That this is a little bit more precise in the sense of actually looks at the disruption of the membrane of the cell. Again, the previous one was more focused in metabolism. There the technique by itself doesn't tell you that the cell has been disrupted. Actually, it's still alive because it's looking at the metabolism, and it's just the difference in the amount of cells. This one looks more specifically that cells has been, or the membrane has been compromised, put it in another ways, and count the cells that are alive, or again, the cell has not been compromised, the cellular membrane, has not been compromised, and compare. Again, you have always a control experiment that has cells hasn't been treated, and you compare with the one that has been treated with your platform, and gives you specific numbers in terms of viability. So those are typical ways to make to measure the cytotoxicity of these nanoparticles. Something that is very, very important, at least from the point of view of therapeutic application, is the mechanism that the cells will use to internalize the nanoparticles. And Dr. Daniela mentioned a little bit about these different pathways of internalization. And why it's important, because depending on the pathway of internalization will be most likely the outcome for where the nanoparticle will end inside of the cell, whether it's going through the endolysosomal pathway or gets directly in the cytoplasm of the cell, or goes to or gets degraded quickly. That's dictated mainly for these endocytosis mechanisms. And I will touch a little bit more in the next topic about these mechanisms with a little bit more of detail without going too much in depth in biology. But here I just wanted to highlight that we study those mechanisms of endocytosis. There are different inhibitors that help us to study that. And again, we use mainly flow cytometry or confocal microscopy, and there is other techniques to study those mechanisms using these inhibitors. Okay. Another technique that we use again, to characterize the performance of these nanoparticles in vitro is confocal microscopy. It's a very useful techniques. It gives you information about whether the nanoparticles has been internalized or not in the cells. I mean, you can see images. What I'm showing here you is different colors in the what we call colors or channels for the microscope for the technique. The blue indicated here is we're staining the nucleus of the cell, and we have other colors here that we are staining either the membrane of the cell. In this case, I think it's more the membrane of the cell, and we have nanoparticles, and we can see that nanoparticles get internalized of the cell. There is a lot of different fluorophores that has been developed for this technique that are very specific for suborganelles or organelles that give us information to let us know whether the nanoparticles are localized in those organelles or not. Again, one of the, at least in my lab, one of the first use of this technique is to confirm that whether the nanoparticles have been internalized in the cells or not. There are certain limitations. This is an optical technique. Certainly we cannot be as precise in terms of the size like TM. What I'm saying is we cannot look at sizes, I think below 100, depending on the instrument, but usually 100 nanometers or 200 nanometers we are already pushing the limits of the optical technique in terms of how precise is that. And I know co-workers, colleagues that are working in more physical, mathematical approaches to push those boundaries. But as far as I understand, optically, I think that's there is certain limitation. Again, TM, you can look, I mean, with precision, nanometers, even bonds. I have seen papers that look at atoms and bonds in TM. And anyway, but for biological applications, you can see nanoparticles in the localized in organelles. Different with TM. I mean, this technique is easy to use. The confocal. What I'm describing with TM is usually cryo TM that requires very specific type of instrument and expertise for preparing the samples. Anyway, so confocal is usually gives us very good information about again localization of the nanoparticles inside the cells and possible co-localization with certain organelles, and you can get pretty images, which is nice also to see your nanoparticles with cells. So that's good. Okay, let me just stop quickly here. Any question for in vitro? that you may have. I'm not sure. I don't have the chat, but if Dr. Daniela see anybody in the chat asking questions, let me know. Okay, if there are not, please feel free to interrupt me. Yes, go ahead.
It is really interesting, actually, and congratulations, because I think your work is really, how can I say, enriquecedor. I don't know what is the word in English, but how can you know in which specific part of the cell is the nanoparticle safe, or yes? Because you mentioned that in some organelles you have seen the evidence of nanoparticles, but I saw some images, so I don't know if in that images some part of the cells are represented in that images.
Yes, gracias, Daniel. Yeah, so let me see if I understood your question. So let's say if we are referring to these images, like for instance, what we are seeing here in the channel, in this case, this particular dye, and I apologize because I don't remember exactly which dye is this right now, but we can see the nucleolus inside of the nuclei of the cell. So we are staining the nucleolus. We're also staining some part of the cell body. And we have nanoparticles in red, then we can see that they are colocalized inside of the cell, and also colocalized with that green dye. This is just I like the images because the cells looks pretty nice. But in particular cases, some of these green dye might be targeting, or might be targeting, let's say, lysosomes. So if those are lysosomes, okay. Here I'm just putting this as an example, and you see that the lysosomes colocalize your nanoparticles, so you can conclude or partially conclude, because it's always to have, I'm saying partially, it's always to have other techniques to confirm that. And partially conclude that those nanoparticles are internalizing lysosomes. Or there are other dyes that are used for the ER, to stain the ER. Or are nanoparticles that are targeted to the nucleus, like in this case, the blue one is a staining dye for the nucleus, and those nanoparticles will be internalized in the nucleus, and you will see the colocalization. So that's the way that we use these techniques for trying to get some idea where these nanoparticles ended in the cell. I can tell you that based on, I mean, it's not easy, it's challenging. Based on our experience, sometimes you will see nanoparticles that colocalize with lysosomes or endosomes or ER. Is sometimes difficult to convolute precisely where they are, and precisely, I guess, will be a key word, because these techniques might not give that precise. And actually, it's a technique that can give you 3D structures of the cells. I'm not presenting here that kind of images, but also can give you more idea to again deconvolute whether it was a lysosome or a ER or an endosome where the nanoparticles are colocalized. But that's the way that we use this technique. Yeah, I hope I answer your question, and I hope I usually tend to give more of the questions, so please stop me if I just keep talking about other topics that might not be part of the question. But I hope I answer your question, Daniel.
Yes, thank you. Actually, I understand more with the explanation of the images in this slide. So thank you, and again, congratulations. It is really interesting.
Okay. Thanks for the question. Okay. Yeah. So if not sure if anybody else has any question or comments. Thank you. Okay. So and again, I just show some of them, couple of techniques for in vitro. Again, there are a lot of more as I show in the list. I just wanted to describe that. I would like to move forward for in vivo characterization. In vivo is important, and more now that if we really want to push our platforms to real application, to real therapeutic of imaging application, in vivo is key to have the basic characterization of those platforms in again, animals will be my term for in vivo. I mean, I can tell you, I don't know, 15 years ago, and Eduardo, when I was a PhD, he visited our lab there, and in vitro experiments were, I mean, are still very important, but you can publish papers with in vitro experiments without much problem. But now, after 10, 15 years now, we can say that no, if you really want to make an impact in the field, in vivo characterization is key. So these are, again, a list of the different techniques basic techniques or experiments to characterize those platforms in vivo. So what is important for a nanoparticle that you want to move forward, again, for therapeutic applications? First, and I might not follow that list, but based on my experience, I will say safety is very important. So the first thing that you want to make sure that you want to know what is the range of concentrations that your nanoparticles is still safe. For that, we usually use mice for this. To calculate the MTD, which is called or MTD stands for maximum tolerated dose, and we tested different concentrations. Again, anything, as I told my students, anything can be toxic. It depends of the concentration. So depending of the platform, it will have more or less a range of concentration, and there will be strategies to actually improve that safety. Now, for cancer treatment, it's really important to know the circulation of those nanoparticles in the blood. What we want is for them to circulate, or at least that's the principle, to circulate, the longer the better, and that's with the target of accumulation in the tumor. So half-life, that is listed there as half-life in the blood is very, very important. And also for safety is the clearance of the nanoparticle. Clearance, and I should say biodistribution, because there are clearance means how fast, shouldn't say how fast, but whether the nanoparticle is cleared by the body or not, and what is the mechanism of clearance, renal or renal excretion pathway or hepatobiliary excretion pathway, and the mechanism of clearance also is related to the time or how fast. That's what I was saying. If it's renal excretion is usually a matter of hours that nanoparticle will be excreted. Hepatobiliary usually take the nanoparticles to different organs, and again, that's important because there are some nanoparticles, mainly inorganic nanoparticles, and size matter, but let's just stay with the type of nanoparticles. Like for instance, gold nanoparticles that most likely won't be degraded and will be accumulated in specific organs like liver, spleen, or other organs, again, depending on the features of those nanoparticles. So it's important to know that, and it's important to know the possible consequences of having those nanoparticles in those organs. Immunotoxicity, again, very key component, as I was mentioned, and I was bringing as an example my collaborator with his siRNA. And even if we have in vitro cells or techniques to give us some idea of how those cells will respond from the point of view in immunotoxicity to those nanoparticles, the ultimate test, well, the ultimate test, of course, is testing in humans, but I mean, in considering the development of the platform, the ultimate test will be to put with animals, because those animals has an immune response, so it will give information valid information about that immunological response. And having said that, and having said about humans, again, bringing my collaborator, which actually, I just keep repeating, my collaborator is Dr. Afonin. He will be presenting, I believe it's on Monday. So you will hear from him a lot about immunotoxicity and immune response from his nanoparticles in our symposium of E4. Anyway, even though the mice has some immune response, that immune response is not the same as humans. Let me just leave it like that. Other type of evaluations that we do, also the ones that histopathology, blood chemistry, those are also related with at least the way that we usually use with the safety of these nanoparticles. And at the end, the in vivo characterization, of course, need to test the purpose of those nanoparticles. If it's a nanoparticle that is developed to have some therapeutic purpose, I mean, cancer treatment, well, we need to have the model, the in vivo model to test that. Or if it's a nanoparticle that is designed to have some kind of, or used as a contrast imaging for a specific imaging technique, well, we need to test that in the model. Or of course, we have the theranostic version of platform, right? That we actually want the whole package there in the same platform. Again, we have the in vitro experiments. Those give us some specific information and useful information. I wouldn't say, oh, let's just move directly to animal models. I don't think I will suggest that ever, because yeah, we get some valuable information with cells, with experiments. I just want to present here the three main type of animal models that we have to test or platforms. Again, these are thinking more in cancer treatment. So we have Synograph models. Synograph models are immunocompromised mouse or mice, because what we do is we inject human cells on those mice. So we don't want that immune system from the animals to reject those human cells. So that's what they are, immunocompromised mice. And those human cells and cancer cells, usually again, is cancer treatment. Cancer cells will start to grow. So we inject subcutaneously those cells in the flanks of the animals, and they start to grow. And what we do, let me just go back to the previous slide, is we measure the growth, the tumor on those animals, and depending on the treatment, of course, what you want is to stop or decrease the rate of growth for that tumor. So those are the Synograph models. We have the orthotopic models. Again, just to reiterate the point, the tumor grows in the flank of the animal. And I think there is this picture is not very clear. This is, oops, sorry. The tumor actually, this is more like in the back of this animal in this case, rather than the flank. But anyway, I just wanted to highlight that because the orthotopic models actually put the tumor where these are still immunocompromised models most of the time. The tumor or the cells, sorry, the cancer cells goes or are implanted where the tumor is supposed to be. Meaning if it's a lung cancer, well, those cells will be implanted in the lung. If it's a pancreatic cancer, those cells will be implanted in the pancreas. And the cells will grow. I mean, the idea is that the cells will grow with the environment associated to that specific organ, rather than the Synograph models, well, that they are just growing in the skin or under the skin in an environment that is not associated to the disease or a specific organ where the disease, which in this case again is cancer, is supposed to happen. Those are a little bit more challenges because a lot of these models, like if you want a pancreatic orthotopic model, well, you have to actually perform a surgery for the animals to be able to implant those cells. So that's a little bit more challenging. And let me just move to the last type of model, again, at least generically speaking, which is the genetically engineered mouse model. So what it happens here, and without me going too much in biology, is these animals are modified genetically in such a way that they generate the disease by themselves. So these are animals we have the experience in the group of collaborating with somebody in biology, and actually I have some slides showing a little bit more about that model, where she modified the animals for them to grow pancreatic cancer. But or the behavior of these animals is such a way that they are not growing the tumors very fast, as in the previous models. And I just go back to the sorry, keep going back and forward. Just wanted to highlight the time here. We are talking here about days. So the tumor already, at least in this model, in 8 days, 10 days already grow in such a way that even some animals already die. In the case of the genetically engineered mouse model, the tumor grow pretty slowly, as happened in humans. I mean, in humans, when somebody gets pancreatic cancer, is not like today got the pancreatic cancer, and in 2 days, 3 days, we have a huge tumor there. In human, this happens in years, in a matter of years. The disease starts to develop pretty slowly, and they're in different stages where the disease goes through until, well, the ultimate stage is when the patients starts to grow that tumor and even starts to metastasize. I guess that will be the ultimate stage. So that was happening in these animals. Try to resemble that progress of the disease. And in that case, this animal takes months actually to grow tumors. And of course, the tumors grow in the organ where they're supposed to grow. Again, if it's pancreatic cancer, the tumors grow in the pancreas in the organ. So it's kind of is what we have closer with all the caveats there with that word closer to what happened in humans for us to evaluate or plan. Okay. Anyway, we have all these different in vivo models. Of course, the easiest to do or to test in the lab is the Synograph model. Again, it's relatively easy to implant the cells there in the skin. The tumor grows very fast. Again, in a matter of days or a month, no more than that, you will have data to have some idea of how your platform is performing. The other models are a little bit more challenging, again, in how they, in terms of time, again, as I say, the orthotopic, well, you need to do surgery. The genetically engineered, you need to have a lot of times. I mean, we have some preliminary data with some genetically engineered model that it took us almost a year to get that data, just because the mouse models, I mean, take that so long or that long to grow the disease. And again, that's the purpose, that they resemble the closer what will happen in humans. And again, the recommendation of the field, based on what I have read in the literature, is, well, try to push to those models, because those are closer to what's happening in humans. And I mean, one of the topics that I will bring later in the next subject is some of the misinterpretation of missed data, missed results that we have get with these Synograph models is specific for the EPR effect, and I will explain later what is EPR effect, what the EPR effect is. But yeah, the idea is to try to use more of these sophisticated models, but to get more robust data, again, with the purpose of moving this platform for clinical applications. Okay. I'm just gonna go quickly through these slides. Again, this is the one that I was mentioning, my collaborator there at UNC Charlotte. So she's developing this genetically engineered model where it resembles what happens with pancreatic cancer. And in addition, for her research, she's actually adding overexpression of a specific biomarker, which is target-specific, tumor-associated MUC1, mucin-1, that she has found that is very important for pancreatic patients. Okay. And let me just go quickly. This is just some use of or some of our nanoparticles in those type of models. I just wanted to present here just as an example of how nanoparticles or these in vivo models are used to test nanoparticles. So this is a Synograph model of pancreatic cancer, where we tested the capability of our nanoparticles to accumulate in the tumor, target-specific nanoparticles. This is a breast cancer model. Also, this particular case is a genetically engineered breast cancer model, where we can see the growing of the tumors that are highlighted there, and the accumulation of some of our nanoparticles in those tumors. So those are the mammary path growing those tumors. And let me just jump to the pancreatic cancer model, where visually we cannot see much because the pancreas is very well embedded in the body. So when we take this the nanoparticle, sorry, not the nanoparticles, the organs, sorry, of from the animals, and we take the pancreas, we can actually see that the nanoparticles has been accumulated in the pancreas. But again, this model gives us the very well characterization in the, again, ideal environment, meaning that the tumor is growing there in the pancreas, and even you don't see huge tumors there, because in reality for humans it's not like they have huge tumor. It's just tissue distributed in different regions in the pancreas that is a cancer tissue or cancer cells that are growing there. So these are the last slides are just some examples from research in my own group that show the application of these in vivo models. And finally, I believe is the last one slide more. But so what is the importance of characterization? Well, is really, I guess, I mean, it seems to be obvious the answer, but it's really important because just from the physicochemical characterization of the nanoparticles and also the in vitro and vivo characterization of the nanoparticles will give you an idea of whether the nanoparticle can be moved forward to clinical trials. Now, what we have seen or what we are trying to push in the last years, at least I have seen that in the field, is to try to standardize those nanoparticles, and that was the purpose of putting this slide that I think Dr. Daniela also showed this slide, is that there are I guess lab entities, government entities that try to give some guidance to have standards characterization techniques or techniques or protocols to make sure that those nanoparticles that are better candidates to move to clinical trials are well characterized, and we are not I guess putting effort, wasting, I guess, time and money at the end in platforms that might not be good candidates for moving to clinical application. And again, also try to standardize the field, because there are so many reports there that show some characterization of those nanoparticles with such a different technique of approaches that sometimes it's really hard to compare data from one paper to other papers. So that's just a personal comment. Okay, so yeah, so this is the last slide for this for this topic. So anybody has a question or comment? Okay.
Thank you very much, Juan. I have a couple of questions, if I can. One is about your in vitro slides, and another one is on your in vivo models. So regarding the in vitro, you mentioned the blood compatibility test at some point. This is something that I very recently started to read about, and I was wondering how common are those tests now in our field, or if you want to look at this in this way, where do you think it is most suitable to start with this blood compatibility, just checking for the behavior of red blood cells and platelet aggregation? Do you think it's something that we should do routinely, or maybe it's for very specific administration, for very specific platforms? What's your view on this?
Well, my view is that we should do it routinely, at least that, let's say, let me just expand this comment. If you want to move your platform for clinical applications, the Nanotechnology Characterization Lab, which is one of the main supports here in US, it will go through all this cascade and maybe more of characterization for your platform. And if it fails any of that, they won't recommend, of course, your platform for clinical application. So my answer will be yes, we need to do that characterization for red blood cells, for hemolysis, and immune response. Now, being a professor, being a PI in a university, I know that our lab has limitations, and we cannot do all that by ourselves, and are still, as I was mentioning before, the field, at least in terms of the literature, is not that strict, which it be. It might change, I don't know. But it's not a strict, like for instance, if I review a paper that is testing a platform, interesting platform for delivering drugs, I might not be that picky as a reviewer to ask that authors to say, well, you need to have some immunological characterization of your platform, because that's very important at the end. Or you might need to have a hemolysis test because that's very important. I mean, at the end, if that platform goes to real application, yeah, that will be very important. But just for what they are reporting in that specific test, maybe just having the MTSSA, maybe just having flow cytometry or other techniques particular for their platform will be enough. Again, at some point, yeah, we again, if that groups want again push forward, that platform has to do those experiments. They have to be there. At the very beginning of the development of the platform, they might not. Yeah, that's my opinion. Maybe I wasn't very straight with my opinion, but but yeah, that's what I think.
No, it's actually clear. I have the same feeling that we are still considering this battery of tests more for regulatory approval than for routine characterization. And I'm also of the view that maybe this is something that we need to still work on a little bit more. My other question was about the in vivo model that you mentioned about these genetically engineered animals. I'm just wondering, are there, apart from time, because they might be very time consuming, I guess those graphs comparing the Synograph with one of these models, when you're testing survival, they will expand maybe for months. I guess that same graph that you have in there, would that be as long as a few months when you have one of these genetically engineered animals?
Yes. Based on based on our experience, yeah, that will be instead of days, will be months.
Okay.
And I mean, this is tumor grow and survival. I mean, I just to bring the experience that we have with one of the papers that we submitted with some of these preliminary data. The reviewers actually asked us to say, well, you should have survival data for your gen model. And what we found is it will take a year and a half of 2 years to get that data. So yeah, so that's way too much. I mean, okay. For my PhD students say, well, no way I'm gonna be 2 years more to here doing that experiment. So yeah, those are the challenges with these models. Yeah, the time and the cost. Of course, the cost also, like, I mean, mice, I'm not sure how many in the audience is familiar, but regular mice cost like $60, $70. I mean, it depends of the country too, but in US, in my experience, these gen models cost like $600, $800 per mouse.
Okay, yeah, that's an issue. But my question was about ethical approval. Is there any extra step that you need for ethical approval? So I'm just wondering, would it be harder to get one of these models approved by the local or national committees?
I'm thinking the answer, because I mean I know it's not. I mean, I know you have to get, of course, more information. You have to bring more information to the ICOOC, to the committee, describing all, because you are doing some, and just moving forward to this slide, you are doing some genetical modifications, so you are having some manipulation there. But I don't think it's something that's, I guess it will require more work, more description of what you are doing with your models. But I mean, it's something that is done. in any, I mean, a lot of universities. I mean, what I'm saying is possible, right? I mean, people does it. It takes more time. Also, it depends of the experience of the committee there, of the IQ committee, because if it doesn't have more experience with us, it might take longer time to review more specific. It does has experience. I mean, I think in universities that has those models all the time, I mean, it might be as easy as any animal protocol. So, so again, I don't see any particular ethic impediment to move this forward. It might just be more work from the PI, the lab that is wanna approve this protocol, and of course I mean, here it will be more the expertise that doesn't have to be much with the ethical part, but anyway, somebody that actually knows how to create these colonies. But no, I don't see any problem for this to be approved.
Okay, clear. Thank you very much.
Yeah, thank you. Okay, let me just switch to my other presentation. I don't have it together. Not sure in time. I think I have taken more than that I supposed to. Okay, just yeah. Please, Daniela, stop me if I'm taking too long, because again, and also my with the interesting questions, I think I'm giving more information that that they are asking me to do it. But anyway, okay. Well, so let me move to the next topic, which are biological barriers for nanoparticle. And it's a very challenging subject because we have barriers all over, I guess, the different aspects where these nanoparticles will go through for this application, for this biomedical application. So I just divided, I guess, barriers at different levels, starting from the molecular level, cellular level, organ and system level. Molecular level, and I will explain more in detail later, but it refers more to the interaction with molecules, those nanoparticles with molecules or proteins, and I guess the other ones are a little bit more self-explanatory. But also what I wanted to mention in this slide is not only that the challenges in the interaction with those different levels, it's also depending on how those nanoparticles are administered, it will have, again, different challenges. So it's not the same if the nanoparticle is going to be injected or administered intravenously or orally or transdermal. They will go through very different routes of interaction with that, I guess, biological entity, whether it's an actual animal. That, I guess, that will be the most ultimate model, right? To whether you inject or administer orally and intravenously, again, will go through different routes in that animal. So you will have to look at your nanoparticle when you are designing a nanoparticle to be able to be stable for all those different challenges. And as I mentioned in the slide, for instance, if you are going through oral administration, well, the gastrointestinal tract has very specific conditions there that a lot of the nanoparticles are degraded under those conditions, and they don't achieve the ultimate goal for the delivery of the drug or any other therapeutic agent. So, like, for instance, that's very particular for oral administration. Intravenous injection has other challenges. So, as I was mentioning in the previous for the previous topic, most of the the presentation of my slides, keep in mind cancer treatment. So most of the nanoparticles that are developed for cancer treatment are administered intravenously. So that's what I will, or the route for intravenous injection that will be described in the following slides, or the barriers for intravenous injection. So at the molecular, as I call it, level, so the first challenge is the interaction of those nanoparticles with the proteins or molecules in the environment. So you start with, in mind, designing a nanoparticle that is going to be beautiful, that is going to be, I don't know, gold nanoparticle, just to bring something that's going to be spherical, 100 nanometers in diameter, well distributed, that you did physical-chemical characterizations, that the TM, DLS, other type of characterization look perfect. And then you say, well, this is the best nanoparticle that we can make. Once you put in a biological media, in cell media, that and mainly the surface of that nanoparticle will change. I mean, one of the features of nanoparticles is that they have a high surface area. So they like to interact with whatever is in the environment. So the first thing that is going to happen to the nanoparticle is again, in cell media, in biological media, is the formation of what people call in the field protein corona. And what protein corona is corona, as is illustrated in these figures, surrounded by proteins. And again, it's just because absorption of those due to the high surface area of those nanoparticles is a physical phenomena that will happen for the nanoparticles. And that protein corona will change the or what people call biological features of that nanoparticles. So now cells are not seen to your gold nanoparticles as shown illustrated here. Now your cells are seen are some entity that is surrounded by proteins. Of course, the core is still gold nanoparticles, but what the cell sees is actually the protein corona. So depending on the protein corona is what the cells will do with those nanoparticles. There are ways to try to avoid protein corona. One of the most common ways is using a polymer, which is called polyethylene glycol, or PEG. So the idea of pegylation is try to avoid the absorption of of proteins in those nanoparticles to keep, I guess, intact the surface features of your nanoparticles. But the use of PEG and the effectivity of PEG is limited. It's not 100% effective in terms of eliminating protein corona. So at the end, you will have to deal, and more not to deal, but to understand how protein corona is either affecting the performance of your nanoparticle, or it might help the performance of your nanoparticle. Again, 10, 15 years ago, the idea in the field was to avoid protein corona, to develop techniques again to make sure that protein corona it was not formed with our nanoparticles. Now the idea has changed. I think people now or scientists in the field is trying to again use that protein corona on their benefit for the application. I was reading I guess a paper yesterday night that was saying, again, 10, 15 years ago, nanotechnology or nanomedicine was fighting with biology. Now, what we are trying to do is using biology to help our purpose, and that's what I'm mentioning about the protein corona. And again, this is just or how can the protein corona can affect the performance of your nanoparticles? Let's say if your nanoparticle was designed to be target specific for certain antigens in the cells, so that protein corona might cover that target group, whether it's an antibody or it's a peptide or it's some small molecule that was used being target specific. That protein corona can and again hide that and avoid the interaction with that receptor. Now, this concept of protein corona is so complicated because protein corona, or the absorption of those proteins in your nanoparticles, is not something static. It's not like, okay, I put the nanoparticle in this media and those proteins are absorbed and that's it. Those are the proteins that will be there during the whole trajectory of my nanoparticle to end in the tumor. No, actually it's a dynamic process where we have hard corona that might stay there a little bit longer, or we have soft corona, and that corona, the features of that corona, the components of that corona also will change depending of where the nanoparticle is, and that's illustrated by the next slide. If it's in the blood, that corona will have certain components. If it's actually internalized by the cells, I mean, the cell has proteins there, and the nanoparticle will interact with those proteins. And again, it's a dynamic process. That's what it's trying to illustrate here. Some proteins will get absorbed, some proteins will get released. And those proteins that get absorbed will be different to the proteins that get released. So that changed the biological feature of that corona. And again, what I was saying is that depending of if it's in the blood or it's passed through the cells or is in accumulated in organs, again, that interaction will make things or that interaction with the proteins will be different and give you a different nanoparticle at the end. And there are labs that are putting a lot of effort, because here you need a lot of effort and resources to actually study this in details. I mean, I have seen reports that where they are looking at hundreds of proteins that are associated to those nanoparticles, and they try to get some correlations of if one particular type of protein is absorbed in more amount than other, and if that particular type of protein is helping to the nanoparticle to get internalized better in the cells or in the tumor. Yeah, so it's a very big topic right now in the area of nanomedicine, from my point of view, to understand and investigate the use of protein corona again in our advantage. Again, now I think that the idea has changed. Rather than try to avoid corona, now it's try to use corona for the application. Okay. So this is just again also illustrating how dynamic is the process, just with some cartoon there, how proteins come and go depending of the nanoparticle, and some of them will be there longer time than others, and that again will impact the ultimate goal or performance of these nanoparticles. And a lot of I guess features will impact this protein corona features coming from the nanoparticle. Again, I mentioned PEG. If you use PEG, you might avoid at certain degree that protein corona if your nanoparticle is functionalized with other type of components, might also impact that formation of protein corona or try to enrich certain proteins to be used in the application. So, okay. Okay, so that's protein corona, and that's what I call the challenge at the molecular level. So now let's go to the cellular level. So in the case of the cellular level, and Dr. Daniela already showed, I guess, a different version of this illustration in terms of the cellular pathways for the internalization of nanoparticles. And these are, I just want to clarify, sometimes we get confused and we think that these are associated to nanoparticles. These are not. These are natural pathways that mammalian cells has developed to reduce to uptake nutrients from outside, or for specific cells to do like macrophage. They use phagocytosis as just, I guess, as an illustration of one of the ways that they do their job. I guess that the job of macrophages is to get rid of foreign I guess material, just put it in that way, and they use phagocytosis to achieve that job. So, and that's one of the mechanisms of internalization of cells. Again, natural mechanisms. There are other different mechanisms which I will illustrate, I guess, quickly here, that those mechanisms are also used by the cells to internalize nanoparticles. Again, at the end, nanoparticles are small entities. That's one of the, I guess, of the big breakthrough when nanoparticles start to use in nanomedicine, is because of the size allows them to be seen as just put it in simplistic ways, as a food for the cells, and that's what they are internalized through in the cells, and I guess we took advantage of that to deliver something, drugs or other genes, other types, just because of the size. Again, cells will obtain those nanoparticles. I mean, what I'm saying is if we don't have nanoparticles, if we have microparticles, most likely the cells will behave different. Most likely, most of the cells won't internalize those microparticles. So we won't talk about the delivery, at least in the way that we see with nanoparticles. So that's what I want to point out, the importance of the size of nanoparticles. So those, can we check? Okay, ya me pasé. Okay. Yeah. So let me just go quickly. I think how many slides I have? Sorry to the interruption. Again, as I mentioned, if I just keep talking about this field, and I just kind of spend the whole day just talking about that. Yeah. So I will try to. Yeah. Okay. So let's see. Toxicology is 12:45. Yeah. I think maybe we move the cancer treatment later. Yep. Okay. Okay. Sorry, I just try to. We try to have some adjust the schedule since it's taking longer than expected. Now why I stopped sharing my, right? Yeah, sorry. Yeah. Okay. Now I'm sharing. Okay. So. Okay. So yeah. So just I'm adjusting some details about the schedule since I'm taking too long with this. Yeah, so I just go quickly through these mechanisms of internalization. I guess just I will go through the basic. If you have questions, let me know. I can go a little bit more in depth of any mechanisms that you might be interested. I will try to avoid too much details. So what I wanted to illustrate here is that depending on the mechanism for internalization, and that's something that I mentioned before, the nanoparticles will be internalized following different routes. I mentioned, well, if it can go in the lysosomal pathway, or it can go directly to the cytoplasm, or other different mechanisms, or other different places inside the cell. So that's what is also important to know these mechanisms of internalization. So. Well, phagocytosis, I think I already mentioned that is used by very specialized cells, phagosomes, and is mainly used to get rid of big material, foreign material, as described here, something around 5 to 10 micrometers, and follow very specific ways for that to happen. And again, I won't go in detail in the biology for that. Micropinocytosis is also another way to obtain foreign material. Macrophages, again, one of the cells that use this mechanism to internalize viruses, other type of materials that, again, they want to get rid of, and they go through microsomes and microphagosomes, and this materials gets degraded, and well, it's just the function of these cells. The main mechanisms that are used for internalization of nanoparticles are, well, let me just say here, there is a lot of mechanisms of endocytosis that people is still studying, or people is still coming with very particular ways for endocytosis of nanoparticles. So what I'm illustrating here are the ones that are being most studied in the field, but these are not the only ones. And again, people whose expertise is in studying the interaction of nanoparticles with cells are coming every year with new ways for internalization of nanoparticles. What is true, the one that has been more studied is the Cabinet-Mediated Endocytosis. Is usually restricted to particles in the size of 200 nanometers or below, is going through a very, very detailed mechanism of how these nanoparticles start to interact with the cell surface of the cells, how that membrane starts to modify itself to get embedded the nanoparticles and form a vesicle inside, how that is pinched off from the cell membrane, and that vesicle start to be internalized. And biologists and other scientists has studied that been very well detailed, as I mentioned. So they know what are the proteins that are involved in this mechanism, and what are the effects or different effects of nanoparticles in terms of size, in terms of morphology of the nanoparticle for this mechanism. The other mechanism is Cabinet-Mediated Endocytosis. Again, this is an invagination of the cells. In this case, the size of the nanoparticles that can be internalized is smaller than that. I believe it's 50 nanometer or below, a little bit smaller. And again, it's a processes that put together a bunch of proteins just to embed that nanoparticles, pinch off that to form a vesicle, and keep moving inside the cells through different processes. These are just summarizing some of those points. Again, depending on the mechanism of features of the nanoparticles or particles, different mechanisms of endocytosis will happen, or phagocytosis. And again, phagocytosis, I want to reiterate that is for specific cells. The same as micropinocytosis is not like like cancer cells, for instance. No, most of the cancer cells, might be some exceptions there, but they don't undergo internalization of material through phagocytosis of micropinocytosis. They usually use endocytosis, and again, that's the importance of nanoparticles for the field of cancer treatment, because they are in the range where these mechanisms work, and that's, I mean, put in a simplistic way, that's what they are internalized. Of course, there are more factors there. Okay, I'm gonna skip this. This slide is talk about the ratio, aspect ratio, morphology of those nanoparticles, how can that matter, and also functionalization of the nanoparticles. And that, again, bottom line, the conclusion is it matters, and that's important because we actually can fine-tune our nanoparticles to direct the mechanism, or to enhance the mechanism of endocytosis of those nanoparticles, depending on those surface features. So once it's internalized, again, it will go through different routes of trafficking inside the cells. The most common trafficking is endolysosomal pathway. Why is important to know this, and why it's important to develop systems that either take advantage of this or try to somehow disrupt these pathways, is because, for instance, for protein, DNA, siRNA, if those nanoparticles get internalized through this lysosomal pathway, most likely those proteins, DNA, will be degraded, which is the function of this pathway, degrade material to get what is important for the cell, and the rest to exocytose from the cells. So if that DNA, that proteins get degraded there, well, there won't be the therapeutic function of those molecules or biomolecules that I think was the intent of that nanoparticle or delivery system. And this slide illustrates some of those conditions in this endolysosomal pathway, usually low pH, a high amount of enzymes. There again, the purpose is degradation. So that's what those conditions. And the field has developed different approaches to try to overcome those issues. Proton spawning phase is one of the most known approaches, where relies in nanoparticles that will abstract, or will let's call it abstract protons from the cell media. in such a way that creates a different pressure or homolytic pressure due to the protons, and will break those vesicles. At the end, these endosomes, lysosomes are vesicles that can be broken using that approach. Another approach that we, I guess other people in the field is looking at photosensitizer to generate reactive oxygen species that also will disrupt the membrane of those vesicles to increase the release of the cargo for the nanoparticles. Okay, so those are just some approaches there for try to eliminate, try to avoid this endolysosomal pathway and enhance the therapeutic delivery of those cargos, mainly proteins or genes. That's one of the ones that are more important to enhance that. Okay. Another, I guess, challenge is to put to the nanoparticles actually in the tumor, and in this respect, a breakthrough that happened 30 years ago was to find out that nanoparticles, different to other materials, has the particular feature that they accumulate in the tumor. And this is called enhanced permeation retention effect, or EPR effect. And that 30 years ago, bring the nanoparticles, nanomedicine to a big boom, because they found out this feature, and actually one of the first drugs that are based on nanotechnology, Doxil, which is a liposome that carries doxorubicin, was FDA approved for clinical applications. And Doxil, at least in the model that was used to be approved, seems to be particularly successful to accumulate in tumors following this EPR effect. So just bringing or linking what I was mentioning with the animal models, EPR effect in animal models, in the Synograph models, is overexpressed, if I can use that word. So you can see EPR effect pretty much in any Synograph models, and that's associated with how fast the tumor grows. So a lot of the nanoparticles are very successful with Synograph models because they are accumulating very high amount due to that situation. But what is what is the simple concept of EPR effect? So tumor, as we know, are cells that grow in a very fast way, in a very fast ratio, different to normal cells. So usually when normal cells grow or organs are developed, of course, blood are the channels to carry nutrients for the cells, for them to grow, oxygen, other type of components. In regular organs, those blood cells are very well formed, usually to endothelial cells, and I'm pointing out here. The endothelial cells are forming those channels, those blood cells, and the endothelial cells are very close together. There is not any gap between them, and everything works as supposed to work in a normal organ. Now, when tumor starts to appear, those cells grow very fast. And of course, also they need to, that tumor, that tissue also need to grow blood channels for their own survival. But in this case, since the formation of the tumor is so fast, those channels are not formed as perfectly as in the case of the regular regular organ, meaning the endothelial cells are not as close as in that case. So there are some gaps between cells, or what people sometimes call fenestrated, that the blood cells are fenestrated, meaning again, they are gaps, and that gaps has the particular feature that allows nanoparticles to pass through those gaps and get internalized in the tumor tissue. So that's again a simplistic way to see the EPR effect. Of course, there are a lot of factors that affect the EPR effect, and not going to go in detail, but that's what was observed and was described as a phenomena that enhanced the internalization of the nanoparticles in the tissue. And again, just bringing back what I was mentioning, well, that's what they observe in animals, and we still observe in Synograph models. Again, Synograph models are the perfect model to represent the EPR effect, and I most likely deals with how fast the cells grow in these models. And the nanoparticle that I was mentioning, which is called Doxil, it was tested in, of course, in human, but in Kaposi sarcoma, which is also particularly, or a model that is particularly representative of EPR effect in humans. So that Doxil got the perfect scenario, or that nanoparticle got the perfect scenario to move forward for clinical applications. Let me just use So what happened in humans, again, and I was bringing this because I will connect that to the one of the last presentations that we have, which is the moving forward this to clinical trials or clinical application, moving forward this platform, is that that people got so excited with nanomedicine 30 years ago, nanoparticles, the application of nanoparticles, that they thought that nanoparticles will pretty much just cure cancer, because it was so successful with this Kaposi sarcoma disease of using Doxil, that people start to dream again the use of nanoparticles for any kind of cancer. Unfortunately, I guess unfortunately for people that is trying to cure this disease or treat this disease, and also fortunately for people that works in nanomedicine, that was not the case. It seems to be that in humans, the EPR effect is not that prominent like in those in vivo models, those animal models, that the EPR effect, of course, is dependent of the disease. And even in the same humans or patients with the same disease, the same type of cancer might present more or less EPR effect, and is particular for that patient. One of the current ideas associated with EPR effect for humans is that testing how prominent is EPR effect in a specific patient using imaging agents, I guess nanoparticle-based imaging agents, and based on that, if the EPR effect is very prominent, to actually recommend the use of nanoparticles to treat that patient. We are now going more through like personalized medicine, because that will be specific for that patient or not. And if that patient, that EPR effect is not prominent, having the treatment with the drug or the nanoparticle, I guess using the nanoparticle might make not much difference than having just the treatment with the drug. So that's where the field is going right now with the EPR effect. And again, is because this effect that we saw in animal models and a specific type of cancer is not that the case with other type of cancer. So pretty much I mentioned some of the concepts that are here about doxil and the Kaposi sarcoma. And this is just an illustration of what I was explaining about the animal models. These are they grow so quickly that is the development of that EPR effect in these models I mean, again, is the ideal models for EPR effect, contrary to what happened in humans, where we have months, years, where the development of the tumor is in the blood vessels that I was mentioning before. That was kind of one of the key components of EPR effect. That doesn't happen. And this is just a slide, just wanted to show Professor Gavison, which was one of the scientists that actually developed doxil, and he is still pushing for alternatives to try to take advantage of the EPR to improve the performance of this nanomedicine, of these platforms. And just to mention, just last thing to mention there, I think this year, Professor Meade, who is one of the scientists, or was the scientist coming with this concept, he passed away. I'm not sure if that makes too much importance for you, but I think that's important to mention since we are talking about EPR. Okay, any question about this? Sorry, I have to skip some slides, and I went through some concepts very quickly. A lot of concepts are more bio related, but let me know if you have any question or comment. Okay, should we move to the next? Okay. Yeah, which is yours, right? Yeah. Okay, so we are gonna move to Dr. Daniela for the next topic. Thank you.
So let's continue with the next topic that is related to toxicity of nanoparticles. I think that we already have like an overview of this topic. For me, it's like the yin and the yang of nanomedicine. Sometimes we are thinking about the good points of using nanoparticles for medical applications, but at the same time, we are thinking about toxic effect of the exposure of these nanomaterials. Is also related to the comment that Dr. Eduardo did before. Is like the new COVID-19 vaccines are based on liposomes. So yes, maybe it will help to the end of the pandemic, but at the same time, it's like something different, new for the population. So what is happening with nanotechnology for medicine? So this is an important concept that I'm happy to present you today. Because sometimes we are just working in the lab and not thinking more about the end life cycle of the nanomaterials that we are developing. Personally, as also Dr. Eduardo shared with us, is like we are there working using organic solvents, using reducing agents, polymers, inorganic salts, and all these kind of things that at the end of the day, we are not really thinking which are the consequences for us and the environment. So that's important to start to take this philosophy of safer by design and greener methods of production. So by definition, toxicology is a branch, it started like that of medicine that will or that deals with the study of the adverse effect of chemicals and other biological agents for human body, but not only for human body, also for the environment. So this is the science that are studying this kind of effects. And if we are talking about nanoparticles, so we have this branch, nanotoxicology, that covers the safety issues related to the use, consumption of nanomaterials. I will go quick in this presentation. I know that it's very important just to have this home message that how we can from the lab apply different methodologies to reduce the risk of nanomaterials. So when we are evaluating the risk and trying to manage, we have to take in consideration different aspects as the exposure or the damage that we can induce to the human health or to the environment. So it's important, all the things that we have been already speaking about, characterization of the materials and how we are in contact with these nanomaterials. I will give an example about titanium dioxide nanoparticles more in the later in this presentation. We will start with the exposure. That is something related with already Dr. Juan Vivero said to us, like these pathways of internalization of nanoparticles, but in terms of like the human body. So how we are exposed to these nanoparticles is not just engineered nanoparticles. They're that we are developing in the lab for a specific application. We know that nanoparticles can have other origin. It's another classification. So we have natural nanoparticles that may come from a volcano eruption or from the space, some dust from the space, or incidentally produced by the human activity. So we are exposed to these nano objects, and one of the paths that are from where we are exposed are the lungs. So when we are breathing, we can have or interact with nano objects. So it's one of the main ways or paths of exposure to nanoparticles. And we will find in our body that we have different mechanisms to protect from the exposure of nanoparticles. So for instance, what we are breathing, so we have an initial layer, as Dr. Vivero said, like to protect. We have certain cells that will recognize what is not normal or natural for us in order to protect and avoid any damage from this exposure. However, if we have nanoparticles, smaller nanoparticles, they can go more inside to the body, into the alveoli, and there, if we don't have this first barrier, these cells that will combat the entrance of this particulate material, so it will comes and we will find another processes like immune processes to fight the internalization or the exposure to these foreign particles, let's say. So if we think about, we are really exposed to nanoparticles through this first entrance pathway. Even when we are cooking, fumes can come and we are all exposed to these. Maybe we are not thinking about this all the time, but it's important to know. which may be the consequences of being exposed to these nanoparticles, natural biogenic particles and biogenic nanoparticles. So it is important. And as we have been saying already, is all these physicochemical properties are very important in how will we react to the exposure of the nanoparticles. For instance, when we are breathing them, there are possibilities of producing oxygen reactive species that can damage the lungs. So if we obtain this scenario, we can even develop some respiratory diseases like asthma, or we can translocate or migrate the particles to other systems. We will see like this consequence. But still there is research ongoing for really understand which are the effects, long-term, slow or exposure, for instance, of nanoparticles. And this is like costly to really understand which is the realistic scenario of this everyday exposure to nanomaterials. We have from the lung, if we damage the tissue, there is a potential possibility of going through another system, and as I said, so here I'm just illustrated with a paper from 2016, how magnetic nanoparticles were found in the brain of people that die in Mexico City and Amsterdam. And authors found that indeed these magnetic nanoparticles are really similar to those that can be developed or fabricated. They found the difference between biogenic and anthropogenic nanoparticles, and they relate these findings to the possibility of developing neurodegenerative diseases as Alzheimer's. As we already say, during the previous session, it's like nanoparticles have this ability, let's say, it can be good or not, depending which is really the idea of the use of the nanoparticles, of producing these radicals and have different consequences at the biological level. So once they are inside, so they can move and damage, and it's an area that is important, or a field that is important to well understand in order to manage the risk of the exposure of the nanoparticles. So this is just to illustrate how really nanoparticles, as the anatomy of the human being, comes from the breathing to the brain, crossing the blood-brain barrier. That is an important biological barrier for the biodistribution of nanoparticles. Another exposure scenario is our skin. So maybe you are familiar with the presence of nanoparticles, engineered nanoparticles, for instance, in different additives or food or for cosmetics. So we have this organ, let's say the skin, that have a particular conformation with different layers. So this is the barrier that will avoid direct contact, for instance, with the nanoparticles. So there are different projects that have been done related to understand how these nanoparticles may cross or not these barriers. So depending on the chemical composition or the classification, if there are organic, inorganic, size, shape, all these kind of things, will directly have an impact on the toxicity that they may cause. So for instance, a European project, Nanotherm, was evaluating the exposition of the titanium, and they were like concluded that isn't a good barrier of the skin, is not depending of concentration and all these things. It can induce a specific effect. So here in this scheme is what I wanted just to show. So depending of the physicochemical characteristics, the composition is how they may penetrate these different layers of the skin. The coating also may have an important role in how they are internalized and may change or induce a damage in cells. So then we have another scenario of exposure is the gastrointestinal tract. So we will see the example of the titania later. So it's a natural way how humans or other organisms takes nutrients from the outside, and depending of the composition of this or natural composition of these organs, so is how they may go through another part of the body, or maybe to translocate to the bloodstream and be eliminated or not. So it is also important to know that depending of the size, for example, in animal experiments, so is how nanoparticles from different origin or different conformation, chemical composition may interact with the human body. In the case of gastrointestinal, path, there are less information, and it's important to know how it have an effect. As Dr. Juan Vivero said, is there are specific also conditions in this system that may degrade or make this barrier for the toxic effect of nanoparticles. Another way of exposure is, as we already said and observed, because of the importance of developing nanotherapeutics, is when we inject the nanoparticles to the vein to be transported to the a specific place that we want, or yes, we want to treat. So we already talked about how they can move through and reach the specific site. And let's just talk about a little bit of this case of a study of the titania nanoparticles. So as I said, we will find this material in a lot of products that we are in contact with during our day, maybe in paints, pigments, or in cosmetics that we use, in the toothpaste, sunscreen, now that we are here in the conference. So are using as an additive for food. So titanium dioxide are everywhere, or in a lot of places, let's say. And we can identify different exposure ways to these nanomaterials for oral entrance, let's say, inhalation from the paints, that if they are not stable in the composition that they have, we can be exposed to, or through the skin, they can maybe have consequences to the human being. So if we think about this management, evaluation management of the risk, imagine this case of the cosmetic sunscreen. So if we have titanium nanoparticles with sizes around 25 nanometers in these cosmetics, so it's because we want to protect our skin from the action of UV light. Why? Because of the intrinsic property that this dioxide has, that will absorb UV irradiation and then no damage will come to our skin or cells. That's the goal of using titania by the property itself. But imagine that we are exposed to this kind of nanoparticles a lot if we are applying the sunscreen. So this is the way of exposure. And even if this is like a relative high exposure to the nanoparticles, there are no risks of fusing or to having these nanoparticles if they are not reaching like internal biological targets. So if we have this barrier, we can say that there is no risk. So there are some studies as the one that I just mentioned, Nanoderm, that was showing this conclusion that the skin is like a well barrier, all these layers, and there is rather a small probability, let's say, to have a toxic effect. So it's not a hazardous nanomaterial for human. However, we have to think about that if even for a human, if the nanoparticles are not penetrated, all these layers in the skin that are part of our skin, it could go out, let's say, and take contact with water and eventually go to the environment. So the effects to other living organisms is something that we have to take into account with this safer by design cycle that we want to evaluate. Another way to be in contact or exposed to these kind of nanoparticles is, as I say, by oral entrance. So as is normally say, this pathway is not like well understood. It's not like easy to reach other organs. So the risk also is small. So there are some studies also telling which are like these normal concentrations per day that we can manage, let's say, for the toxicity. So as few additive, as I said, there are few studies, and there is any alarming effects of this exposure to titania nanoparticles. Indeed, if we see in this nanotoxicological letter, this article from some years ago, we can compare a little bit like the exposure of titania to the consumption that we have with common salt or that we use to cook. So it's not far from it. And indeed, we don't think that we are in a risk when we are adding salt to our meals. So it's just to compare in the case of this pathway of exposure. So there are some principles that are involved in nanotoxicology. I will go fast in this part, as we have been already spoke about, that have a lot of similar things like with the physicochemical characterization of the nanoparticles. So the first principle is the transport one. So we already talked about the different uptake mechanisms that nanomaterials have at the cellular level, for example. So depending of this and where the nanomaterials finally localize, so is the toxic or not toxic effect that they may induce to the body. We have here an example of zinc nanoparticles. We know that zinc is like an element that sometimes we are taking to enhance the immune system response. Is very common that some of us, we are thinking this additional thing of what we have. So imagine or consider that the nano design of a zinc oxide, it's very important to be considered, as it's not the same quantity of a conventional zinc presentation that the quantity of the element that we will have in this nano building agent. So it's important in the transport of the different zinc, let's say, conformation. So I'm just presenting two examples here, or illustrating here two examples that I found, like zinc like nanoparticles, and they are in the market, and people use them for as additive or for nutritional things. But it's important to think about the concentrations, as it's not the same this building of the nanoparticle that alone. So here is liposomal, and here is like nanoparticle. I need to check more in details this, but it's really interesting to know that all this is in the market, and we have to know which are the toxic or potential toxic effect, if there are. Then a second principle is about surface. We have been speaking about surface. Maybe I didn't say like precisely when we were talking about the texture of nanomaterials. So depending of this parameter is the surface area that we have, and if the surface area of nanoparticles is high, so will enhance the reactivity, let's say, of the material. So if smaller the particle, higher the surface area, and thus we have more possibilities to interact with biological matter, and then maybe to induce a cascade of processes to or with toxic effect, let's say. So this another principle that have been considered for nanotoxicology. And finally, the material. So it's particularly the part of the nature of the nanoparticles. Again, size, shape is all taken into account, but even if we have, for example, here carbon in different conformation, so we will have different effect in terms of toxicity. So all this together, surface, material, will have an effect in the toxicity. Finally, another part that is taken into consideration when we are talking about nanotoxicity is, as I say, like the effect that we have in the environment. So again, is we will find different ways of exposure or scenarios. Let's say in the salt, water, and air, we will find natural or anthropogenic nanoparticles. And what is the potential risk for the population or for the living beings in the specific places. So it's something that nanotoxicology is looking for, is like to take the data and to really analyze which are the risk to manage this. But still there are like a gap in this kind of evaluations. That's why I will emphasize with the 12 principles of green chemistry to reduce or to eliminate the release of contaminants and all these kind of things when we are doing activities as us developing nanoparticles, for instance. So this effect can come to different places, and is something to consider. I'm I was very fast in this part, but it's something that we have to take into consideration. So I would like to know if you have any comment to this part. And I think that's all for me. I cannot see them.
Thank you very much, Daniela. Juan, I just wanted to leave one thought with all of you here. It seems to me that nanomedicine today, it's going to be a very tightly regulated field. So we need to demonstrate that what we are doing is perfectly degradable, or it's not going to be accumulated or to cause any big toxic effect for humans or the environment. But also what Juan was mentioning before, it looks like partially a failure that we were not able to use the EPR effect and to do the accumulation in the human body to treat diseases. So it seems to me, and I just want to know if you agree with this thought, that altogether, so these difficulties in the field that we need to demonstrate a lot, but we failed somehow to demonstrate a clear benefit or a quick benefit for treatments such as cancer. Is this one of the reasons why now it's harder and harder to get funding to continue with nanomedicine research and to propose some new systems, and then reviewers come back very quickly saying, no, this is not realistic, you need to think in a different system, maybe decrease the level of risk? I don't know, I have this feeling. I would like to know what you think about this.
Thank you, Eduardo. For me, as I want to say something. As young researcher, is like very important to think about the whole scenario of developing this kind of nanomaterials. I understand the benefits, but it's not easy to show these benefits to the population, let's say, or to the authorities, or here in Mexico, it's like difficult to consider all these kind of things. And for research, we have to take into account a lot of things, as you say, maybe. That's why sometimes it's complicated to be founded in this project. But something that I want to emphasize since also the beginning is this interdisciplinarity of nanomedicine. So even if we are just looking or evaluating the biological effect in vitro or in vivo, if we are collaborating and we can like see the huge or not the huge, the entire scenario and evaluation of these proposals of using nanoparticles for medicine. Maybe sometimes when I'm speaking English, my ideas are not coming like clear as when I'm thinking in Spanish. But for me, it's, I understand your point, and also I'm trying to imagine myself, and what I want to share with the new generations is like we have to think about the design, but not like a piece of thing independently of the entire cake. We have to see everything, not just the piece, but yes, we can work in the piece. I don't know if I was like clear with this analogy, but it's challenging to work in research in nanomedicine, let's say.
Yeah, yes. I just want to comment. That's, I mean, certainly I agree with you, Eduardo, and and with Daniela, I mean, in the sense that, but with Daniela, in the sense we need to look at the big picture of the application of nanomedicine. That sometimes is hard because we are focused on a small, well, small just for saying some fields or application of the platform that we are doing. And with what you mentioned also, I mean, I agree in the sense with the EPR. I guess my vision there is, I'm not sure if I will call it completely a failure. I mean, in the sense that, I mean, the APR is just an effect or a phenomenon there that was discovered. The problem, what I see is that was oversell. I think that's the main problem, that the field nanomedicine and nanotechnology in general promised too much at the beginning. I remember my advisor, Victor Lin, saying, well, just put nano in the proposal and it will be funded. That was 15 years ago. And seems to be, and I agree with you, now you put nano and the reviewers are looking with more detail whether what we are proposing makes sense or not. So yeah, certainly the field went through a big boom with new discoveries, a lot of promising, and we were getting a lot of funding. Now it's going to go down in terms of what people believe that is delivering. But what I feel is with, for instance, with these vaccines, with COVID-19 vaccines, with some of the Pfizer, Moderna are using nanotechnology, nanoparticles for the delivery. I think people might still get a little bit more hope, or the hope back with nanomedicine, the application of nanomedicine for, for different diseases. But yeah, certainly it's a tough time now for people that are working on this in this field.
I agree. Thank you very much.
Okay, so let me just share. So the idea is to have one last topic for us to discuss, and I think I did I share? Yeah, I share. To discuss, and and we have a lunch break, and just we will have 3 topics more to go for our tutorial. Yeah, has been a long tutorial, but again, I hope that it's been helpful for you to understand some of these topics, and we really appreciate the discussion that we are having. So cancer treatment, and it's really a hard topic to to go through, because a lot of the nanotechnology that has been developed with medical applications is related to cancer. Again, just bringing my previous comment with the APR effect, what we were just discussing with Eduardo's comment about the first, the original promise of this technology to cure or treat cancer. So since 30 years ago, a lot of was focused on that field. Now other diseases, or the use of nanotechnology for other diseases is catching up, and honestly with more success that the application of nanotechnology to cancer. But anyway, we have a lot of information and a lot of papers published in this field. So just focusing on or trying to give a talk about the nanotechnology can be overwhelming. So that's what I make it simple, hopefully simple. And I put one example from home, from my own group at the end, just to show some strategies to develop platforms for cancer treatment. So I found this review, which I have the reference in the previous slide from ACS Nano, pretty interesting and simplistic to the way to illustrate the strategies that we need to pursue if we want to design platform for cancer treatment. So as I was, I guess, mentioned, part of the ideas with this barrier, biological barriers for nanoparticles. Well, one of the points is of these nanoparticles is to go through the blood, being most likely intravenously injected. I guess it will be the way backwards. I started with cells or with protein corona, I guess. Anyway, it will be intravenously injected, circulated to the blood, optimize the circulation to the blood. Of course, try not to make it safe. Don't create any aggregates that might have issues in the lungs or other organs. And that circulation in the blood will help, hopefully, to improve the accumulation in the tumor. And once it's in the tumor, will penetrate in the tumor and penetrate in the cancer cells. Something that I just quickly mentioned, and I'm not addressing, we're not addressing in this tutorial, but I mean, it's important. Again, we decided, or we have to decide what to put or what not, because of the time, is that we usually see tumor tissue as a very simplistic way, that in the sense that it's only made of cancer cells. And the nanoparticles get there, cancer cells are there, and the nanoparticles will reach the cancer, the liver, and kill the cells. And there is not that the case. A lot of the tumors, they have what people call stroma or tumor microenvironment, which it means that a lot of that mass that the tumor is, or the composition of the tumor, is not only cancer cells, is other type of cells, fibroblasts, which are very specific, are called tumor-associated fibroblasts, certain times that macrophages, there are collagen, biomolecules like collagen, and other type of biomolecules or other type of cells. So in reality, that mass of tumor is I guess a universe of different constituents that goes from cells and biomolecules, not only cells. And I can mention that, for instance, in pancreatic cancer, the amount of the percentage of pancreatic cells, or sorry, the cancer cells, pancreatic cancer cells in that tumor is around 20%, 30%. So it's not only that the nanoparticles to get there, but also to localize the cells. And again, opening this parenthesis just for you to have also that concept, even I'm not illustrating with more details. So just for us to know that, again, even reaching tumor or penetrating tumor doesn't mean that the cancer cells will be immediately there. So there are other constituents in that tumor tissue. But anyway, going back to to the route for the nanoparticles, the route that will follow for cancer treatment is, once they reach the tumor and hopefully localize localize the cells. I mean, localize means that the nanoparticles get internalized, interact with cancer cells. They are internalized, and well, it will release the drug. And here we have all those barriers that we're mentioning before. But that's the, I guess, the whole journey that these nanoparticles will undergo for cancer treatment. So the idea is to develop nanoparticles with particular features, physicochemical features that will allow to maximize that process. Yeah, and I find this I guess cartoon or chart pretty helpful in that sense. So if we have, for instance, and they define, the authors of this review, they define very specific features for the nanoparticles, like drug retention or release. They call it 2R, surface is stealthy versus sticky. They call 2S, tumor penetration. So they call this the 2R, 2SP properties, and also stability that are associated with, and the properties that are associated with stability, surface, and size, what they call the 3S properties. And they relate all these properties that they define with, again, that journey that I just described in the previous slide. So if we want to, we need to design we want an optimal nanoparticle for delivery, for cancer treatment. We need to design a nanoparticle that has a very good retention all the way through the circulation, penetration, internalization, all the way until the nanoparticles get inside the cells. There, you don't want your nanoparticles, your drug. There, you want your nanoparticle release the drug. So the idea is to think ahead mechanisms that will allow that specific release of the drug just right inside the cells. And why is that? Well, as you know, one of the main issues with, at least with chemotherapy, is the side effects of those drugs that, because they are not specific to target the tumor, they go to other organs and they produce issues. I mean, all of the issues that we know of, that people has with undergo chemotherapy, losing hair, and other, I mean, problems in immunocompromised system are associated to that toxicity of the drugs because they are not target specific. So if we don't design our system that is retain the drug, is pretty much it will be performing as just injecting the individual or the free drug. So that's what is important, retention in all these steps until it reaches or is internalized inside the cells. So this concept of stealthy is associated with what I guess I mentioned with it's related to the protein corona. I didn't go in depth about one of the issues with the nanoparticles that they are covered with the protein corona is that they are recognized by the immune system. Some of those proteins most likely will trigger the immune system, and the immune system will call macrophages and get rid of those nanoparticles. So we want to design nanoparticles that are stealthy, like invisible to that immune system, to avoid these nanoparticles to be removed from the body. And again, all the way until we get the cancer cells, because now in cancer cells, we actually want those cells to internalize the nanoparticle. And the idea of penetration, well, that happens mainly in the tumor. So stability, again, we want a nanoparticle be stable all the way in the journey until it reach the tumor site and the cells there. We want the nanoparticle to be degraded. The ideal system, I mean, it's pretty much just described that, have a nanoparticle that hold the drug without releasing any drug until it reaches the tumor, get to the cancer cells, and there release the drug and also degrade itself in biocompatible components that can be excreted. That will be the ideal system, and that's kind of what happened with liposomes. The main, liposomes in the part of degradation, there are still some issues with the stability of liposomes, as far as I understand, in the process when they are injected in the cells and on all the journey until they get to penetrate the tumor. But the big advantage of liposomes is when they are internalized in the cells, the way that they release the drug is actually the components of the liposome embedded with either the vesicles of the cell membrane, because at the end, the lipids are natural lipids, or most of them are biocompatible, and they stay there as part of the body without any issue. So that's what we want, and that's ideal, or that's ideal. For other platforms, like what some of us that we are working with inorganic nanoparticles, we try to develop systems that under certain conditions inside the cells, those nanoparticles start to degrade, not only to release the drug, but also degrade themselves to hopefully take advantage of some of the clearance pathways that the body has, to make sure that there is nothing behind to avoid any toxicity. Well, in terms of surface modification of the nanoparticle surface, again, pegylation will give you a stealthy, as I was mentioning with the protein corona, or at least that's one of the ideas for nanomedicine. Shielding functional groups and also the nanoparticle to be neutral, it will help to make sure that those nanoparticles are not recognized by the immune system. But once you get to the cells, again, you want the cells to recognize the cancer cells to recognize those nanoparticles to be internalized. At some point, pegylation might not work because you are avoiding the interaction with the cells. Cationic charge is ideal because just electrostatic interaction with the negative charge of the cell membrane, that will help a lot to enhance the internalization. Again, but cationic charge is not very specific. So you have to be there in close proximity to the cancer cells for that to work. And size, again, usually 100 nanometers is still kind of the criteria for accumulation. Penetration, some recent report has looked at nanoparticles lower than that. I was more in the sense of 50 nanometers. Here, these authors are mentioning less than 20 nanometers. So I guess putting together a nanoparticle or with all these features is challenging. Scientists in the field are looking for different strategies, are looking for, just to mention one, for instance, pegylation. The pegylation, they are looking to attach PEG through stimuli response linkers that once they are internalized, nanoparticles internalized to the tumor, under specific conditions due to the tumor, those linkers, they are broken and PEG is PEG is removed from the nanoparticle, and that will have the interaction with the cells, and they also put some positive charge there to show that positive charge in that steps and enhance the internalization. Anyway, scientists in the fields are using different strategies for for hope with the hope not with the hope, but the designers, I guess, smart designed these nanoparticles to try to follow those rules that were described before. That again, some of them are contradictory, and you want a nanoparticle that is stable for the drug, not release the drug, but suddenly at the end you want a nanoparticle that release the whole drug for the therapeutic effect. So these properties that we want in the nanoparticles sometimes can be contradictory in that way, but chemistry, engineering, material science, I think have the knowledge or the techniques, chemical reactions, take advantage of chemical reactions to develop stuff like those systems. And there is a lot of platforms that are out there that have been explored to accomplish this goal of delivering drugs for cancer treatment. I just put some of the ones that are the well-known in the field. Again, every year new platforms come up to try to again to reach this goal or achieve this goal of developing better systems for the treatment of patients with cancer. Let me just, I just want to mention that right now the systems that are, the platforms that are more approved in the field, at least in terms of clinical applications, are liposomes, as I was mentioning, and polymeric nanoparticles. There are other developments based on organic, or what I call organic nanoparticles, or also inorganic nanoparticles that start to move forward to these clinical applications, but at very slow, it's very slow pace. Okay, and I just wanted to illustrate some of this concept. I mean, with an example from the house, from my group, using mesoporosilica as the main platform that we use in my lab, at least for chemotherapy. In this case, we use this platform. We try to take advantage of this platform to release two different types of drug, which is called cisplatin and gemcitabine, and to treat pancreatic cancer. We also use our collaboration with Dr. Mukherjee, taking advantage of an antibody to target tumor-associated mucin 1, which is overexpressed in pancreatic cancer. So the design of the platform is shown here. We wanted to develop, as I mentioned in the previous slide, a system that will be stable under, I guess, those conditions or those different steps in the route for the delivery system, meaning that we didn't want these drugs just to be physically encapsulated because they might be leaked from the nanoparticles. So we wanted to use chemical bonds that will allow to tightly contain the drugs, both cisplatin, gemcitabine. And one of the features of these chemical bonds, we use stimuli responsive linkers, in particular disulfide linkers for cisplatin, for gemcitabine, and cisplatin also responds to redox conditions. So both systems, both nanoparticles are attached with linkers that will be broken in the reducing environment. And we are taking, or the idea is to take advantage that they are released inside the cells when in a high reducing environment, and also as is known for cancer cells and in in the cytosol or in the cytoplasm of cells. So that's the way that we are having our control. That's the way that we are having our retention for the drugs, and also release. And the hobby is release inside the cells. We use pegylation to give this stealthy part of the nanoparticle, and we use an antibody to hopefully make it sticky once the nanoparticles reach the tumor, to be more target specific, or be target specific to the pancreatic cancer cells. So this is, again, kind of applying those concepts to this platform as an example. This is just a quick photophysical characterizations. I guess I'm not going in much detail. Nanoparticles are around 40 to 50 nanometer average in diameter based by TM. The hydrodynamic diameter shows nanoparticles around 100 or so nanometers with okay, I guess, distribution based on the PDI. We tested these nanoparticles in vitro using optimized ratio of the drug, and we can see the synergistic effect of our nanoparticles, again, pancreatic cancer cells, and the optimal, well, not the optimal, should I say, we use the word optimal, but the better internalization of our platform in cells that overexpress the TMUC1 antigen, that the antibody that we attach to the nanoparticles will enhance that internalization, which we proved that that's the case. These are images I think I showed in a previous presentation, but we use a syngeneic mouse. I guess I didn't explain what this animal model means, but in this particular case, this is like a Synograph model, in the sense that these are cells that are implanted in the flank of the animal, but different to the Synograph. This model is not immunocompromised. It's what people call immunocompetent, because the actual the cells that are implanted comes from another mouse, the genetically engineered mouse model. So anyway, the immune system is intact for this animal, and we see localization of our nanoparticles. There we have control experiments like with the nanoparticles without the tab, so we can see a higher internalization with the nanoparticle that has the antibody as an indication that indeed the antibody is doing the work. These are data for the therapeutic efficacy of the nanoparticles. We can see, compared with other control experiments, that our nanoparticle that is targeted with the antibody has a better therapeutic response, corroborated by the tumor weight. And again, these are just data results showing that applying those concepts that are mentioned at the beginning of the presentation can help to design a system that can be effective. Of course, I'm not saying that this is the perfect delivery system, but it's certainly working, at least on the models and the data that we apply this platform for these models. We also corroborate that enhanced therapeutic effect using immunohistochemistry. And in terms of the safety, we can see that our models are safe, but using H&E, we actually found that the free drugs has some impact on the kidney, which is not surprising. Cisplatin is associated to, is a very nasty drug in terms of the side effects of kidney for patients that are receiving, and actually that's some of the reason why cisplatin hasn't been used that much lately in clinical applications, or has to be reduced because of the toxicity. But we can see that our nanoparticles actually reduce that effect. So that's kind of the other beauty of nanotechnology or nanoparticles for cancer treatment that it reduce the side effects. And again, I just wanted to illustrate with this example some of the rules that were mentioned in the review of how these concepts, and again, those concepts has been built in decades. I mean, it's not something that we knew 30 years ago that, okay, those are the rules that we need to follow. And most likely in 5-10 years, if we are still working in this field, most likely some of those rules will change or new rules will appear for the design of these systems. But I mean, based on what we know so far, I think that's a good way to go. And at the end, that's what science is about, right? We always learn something new, and we keep advancing the knowledge. So that's what I have for this topic. So do you have any question or comment about this? Again, I try to describe some of the main rules for designing systems, nanoplatforms for cancer treatment, and I just I guess showing something from my lab just to further illustrate. What? Oh, thank you. Thanks a lot. If there is any no questions or comments. Okay, so I think the idea is to take a break, right? A lunch break. So we really appreciate that you have been with us this Sunday. I know I'm not sure if it's the dream of everybody to spend a Sunday talking about nanomedicine, but we really appreciate that you are holding there your comments, your questions. And yeah, so the plan is to take a lunch break and come back at 3. Is that correct? Come back at 3 p.m. So please come back. So there are 3 more topics to cover. Yeah, to close the tutorial session. Okay. Thank you. We will see you at 3.
Thank you, Doctor.
Thank you.
So thanks again. And this part is application of nanomedicine for biomedical imaging. So this topic related to the use of nanoparticles for diagnosis, and some of them with this ability to be used also for therapy in when we are talking about the diagnostic. So this is like the big picture of how nanomaterials can be used or applied in vivo imaging of what can be translated to medical imaging technique. And in a specific way, it will depend of the properties of the nanomaterial, the technique that we can use to have this imaging. For instance, we can find magnetic imaging using nanoparticles that have this property to react or to be excited with a magnetic field and come back to the to a stable state, and in this way to have like an image. Then we have another way to of applying nanomaterials in imaging when they have optical properties that they may be luminescence, fluorescence, or for instance, when we are talking about metallic nanoparticles, this plasmonic effect that induce the possibility of image and therapy, resonance energy, or photoacoustic properties. Then we have what is known as nuclear medicine techniques that will react or interact with ionizing radiation and have different phenomena presented in the nanoparticles to have an image. And others acoustic, when we have a light that is applied and then sonic waves that can be detected to have the image. And something very interesting in the way of use nanomaterials for in vivo imaging is that, again, we can play with the design of the nanoparticle, the surface chemistry, to have multimodality in these nanoagents, so we can modulate the properties and the response to for instance, incoming signal to have a detection and then use it for imaging. So normally when we are working with nanomaterials with potential application in medicine, so it's important, as we have been already discussing, that use in vivo models, animals, that will allow us to have this proof of the concept of the imaging and then the safety of using the materials to have a clinical translation, how to use these new agents for giving contrast to the final image. So there are two possibilities, let's say, or how these materials work as contrasting in imaging. So we have, for instance, when we have an input or an output signal, so depending of the intrinsic properties of the material, we can stimulate it or excite it and then collect a different signal to produce an image that will give us information about the localization of a tissue or the tissue that we want to image in a selective way. Or just when we are using or detecting an output that is not necessary to excite it by itself have the property of induce or to present something that we can detect to form the image. So nanomaterials, here I'm giving an example of how also these physicochemical properties can be modulated or modified in order to maximize the outcoming signal that we need for the image. Here we have the example of 3D structure of or gold nanostructure when we have nanorods, 2D. When we excite these nanorods with light, we will have, or they will present some phenomena that will hit locally the tissue where they are localized, and this thermal property allows them to go back to initial state, and by the expansion and this go forward to the initial state, we can detect ultrasound signals. So this how we can use the nanorods for for this kind of modality. We are just, for instance, here presenting what is the way we can use as contrast agents the nanorods. Nanomaterials for these applications. So we go back to some classical characteristics that we can find in the nanomaterials. So we know the dimensions. So we already know also which are the advantages of using this scale, dimensional scale. We can work with a surface to enhance the binding to different tissues or specific sites to, for instance, diagnose a tumor. We can modulate all these physicochemical properties. That's why nanomedicine is so flexible, using different synthesis and functionalization techniques. So we can control the size, the shape that determine some of the optical or magnetic properties, the nature of the material, the quality that we have, the density in terms of the concentration, or also in terms of electron density that are needed for certain imaging techniques, surface charge, as we say. We can load some of these designed nano tools for delivery. In this way, we can apply different modalities of treatment. For instance, we have done this, as I say, this capacity of combining different techniques. In the picture, we are seeing an example of an iron oxide nanoparticle that have different coatings or functionalities in order to have multimodal imaging diagnosis. For instance, if we are using a fluorophore organic molecule that have optical, in this case fluorescent properties, or another entity that we have that will present a property of being detected for nuclear medicine with a chelate using a radioactive species, and then all the things that will be needed, or the components that we need in the surface to give some specificity or stability for the applications in vivo. So depending what we are, let's say, exciting, or which kind of or modality of imaging we want to detect, so we enhance the contrast of the in vitro imaging. In the first part of the image, in the section B section, we have at different times how this kind of nano tool is internalized, biodistributed in this animal model by fluorescence. So when we are using this fluorophore, and in C we have the image that we obtain when we are practicing PET imaging, that has this that detect the deposit transmitted by the radioisotopes. So when we are developing this kind of nanotools, is the goal of diagnostic agents that we can, for instance, treat or at the same time with a single agent have the diagnosis and the treatment of a tumor. We have also this possibility of the passive targeting by the APR, and why not the active targeting? We want an accumulation and to enhance the selectivity of the vectors that we are developing. So it is important to to have an idea, to know which are the strengths of each modality in order to design, synthesize, characterize the nanotools that we can propose for this purpose of imaging. And something very important is the advantage of nanotechnology that we just discussed, that we can combine different imaging modalities in one structure to improve indeed the diagnosis and the therapeutic index of the different combinations of modalities. So here we have an example of nano development that use also a radioactive marker or radioactive element that have a specific lifetime that will allow us to use positron emission tomography imaging that is classified in this nuclear medicine. And by using or by functionalizing the nanoparticles, we can add what we say like molecules that would give properties, some circulating properties or others. So it's different ways or designs that we can use to combine the imaging modalities. In the table, we are just presenting like a summary of some of the main concepts that we have to take into account, or that we have to have an idea of if we want to develop nanomaterials for different imaging modalities. So we know which are, for instance, the modality when we are taking into account the magnetic properties of nanoparticles or nanomaterials in general, which are in these strategies the input signal that we need, how we can excite the molecules or the nano-constructed structures, which are the temporal resolution, the depth, in order to get an image of the tissue and diagnose maybe a tumor site. Then, which are the imaging modality when we are thinking about optical properties of nanoparticles. So, which are the quality of the radiation that we need to excite, and then to detect another signal. So for instance, we can use ultraviolet to near infrared light, but it will depend of the localization of depth of the tumor, for example, to use these techniques, as we are not in the range of ionizing radiation. So we can find here all the characteristics that we can take into account for these nanotools for imaging. We were saying that there are other phenomena like acoustic nanomaterials that can be used when we apply sound waves or first light, and then to detect these waves, or nuclear materials for gamma imaging or positron emission tomography, and others like computer tomography that use X-rays as input source of energy to give origin to different interactions with matter. And depending of the facilities that we have, obviously we will see an impact in the cost of the diagnosis modality. So I will just talk about briefly about different modalities of imaging, starting by computer tomography. So as I just said, is the use of X-rays to image tumors. So it's not an invasive technique of diagnosis due to the wavelength of X-rays. So it's a high resolution technique that allows imaging anatomic scale, and depending of the orientation of the beam, incident beam, so we can even construct a 3D imaging. We have a low sensitivity. It requires also the administration of a contrast agent. Which are the characteristics of this contrast agent? So high atomic number. It is important to have this interaction with X-rays. So electron dense objects or atoms are important for this technique. Which are the disadvantages of the use of contrast agents for computer tomography? So sometimes we need high concentrations of the contrast agents that are, some of them, estaba con eco ya. Sorry, that the high concentration of the contrast agents of iodine or of barium, that are the conventional ones that are used in clinics, they have, due to its origin, some potential toxicity, short blood circulation time, and as single molecules, they are not selective for the imaging. Now, if we think about the use of contrast agents, but like nanomaterial, so we can overcome these limitations of the technique. We know that we can change the size, shape, and composition of the nanoagent. There are we can load it with different high density atoms, for instance, gold, or different from the common molecules used. And we have an enhanced contrast by applying nanostructural materials. This is an example by comparing the use of hafnium and zirconia metal organic frameworks for computer tomography. So if we are designing contrast agents for computer tomography, so it's important to, as I say, consider the atomic number. So just briefly, I would like to show in this graph, for instance, the value of the contrast as function of the concentration of these two MOFs that have been built with hafnium zirconia and have like a silica shell to give more biocompatibility to this structure, and PEG for these effects that we already know for the circulation. So if we observe then the graph, it is a clear evidence that by using a heavier element, we have a better response in the contrast. Hafnium must have like more than twice the weight of zirconia. So zirconia is 30, something like that, hafnium 72. So it's very important how the atoms may interact with this ionizing radiation, the X-rays that are used like an input signal. And do not forget that when we are developing this kind of materials, so we can have all the surface chemistry needed for safer use of the agent. So in the images that we are here, we are focusing different places, organs, and in the tumors, so the contrast must be higher in the site that we want to diagnose. Another modality, as we said, is magnetic resonance imaging. So this, generally speaking, is again a non-invasive technique. So we are not opening or doing something to the patient. It's a non-invasive technique. We obtain a high resolution, again, anatomical imaging. You can observe the organs and all these. Low sensitivity requires also the administration of this kind of ions to enhance the contrast of the imaging. Disadvantages of using this technique for the diagnosis is also the necessity of use high concentration of the contrast ion. Some of the conventional are based on gadolinium and iron that present the magnetic or paramagnetic properties that we require for applying this technique. And we have, for instance, also the limitation of some potential toxicity if, for instance, the patient that we want to diagnose with this technique have renal dysfunction, short circulation time. With these chelates, we cannot be too selective as by using nanotechnology. Low responses to the magnetic field or relaxation properties. And now if we imagine the scenario by using nanostructured materials for this application, for this modality, we have the same advantages naturally coming from the nano science for the nanoparticles. So regarding the physicochemical properties that we have, they are positive contrast ions. We can increase the gadolinium or the magnetic centers. And we obtain by this superior, let's say, enhancement of the imaging, which are some of the disadvantages that we can consider by using these nanoparticles is slow excretion after the imaging examination, long-term tissue accumulation, and maybe toxic effect of the ions, of the gadolinium ions that are those that have the magnetic properties, some fibrosis or problems with the tissue. And now I will we can see, for instance, an example by using mesoporous silica nanoparticles. Indeed, this is a work that Dr. Juan Vivero Escoto developed in 2011, if I'm not wrong. So it's the use of these nanostructures like efficient platform to develop materials to enhance the contrast properties for MRI imaging. So here is a scheme from the work that he developed with his group. So in principle, it's something similar, let's say the conception of what he presented just before the lunch break, but it's the use of silica mesoporous nanoparticles that, as I explained at the beginning, in the morning, is by using chemical techniques, we can really control the texture, the size, pore size, and all the physicochemical properties to encapsulate or to put inside the internal domain some molecules, and then in the outside other linkers that we allowed, and to give this specificity to the vector. So it's what is represented in this scheme, like how we can put inside the the nanopore some functionalities, in this case gadolinium chelate, that will have this property of magnetism. Then how, depending of this bound, disulfide bound, we can have a smart nanomaterial that will respond to a natural stimuli that we'll find in cells, for instance, or redox reactions can come here to break and then deliver selectively when we are using different ligands in the surface, the contrast ion that we need for the imaging. So here, just to be more clear, we are the scheme is representing how polyethylene glycol has been used to give this colloidal stability and this effect that we need for circulation and accumulation in the tumor, and how is conjugated with a target molecule. So this is a possibility or an alternative that have been studied in his group for MRI imaging. So here's some of the presented results when they take into account with a comparative purpose conventional contrast ion, and how the different conformations or the different formulations that they develop show different contrast properties at similar concentrations. So they observe that when they have the complete system, let's say with polyethylene glycol, they have a better response in the contrast of the image. And something important also in the flexibility of this kind of nanovectors or nanocarriers for contrasting agents are the possibilities to use optical techniques at the same time or with the same vector, like with the magnetic properties. And just briefly, I'm presenting here what they published, that are confocal microscopy images, where they were labeling the mesoporous silica nanoparticles with a fluorescent dye, and they observe, for instance, the internalization in cells. And again, depending of the surface chemistry of the particles in pancreatic, that is the model, pancreatic cancer cells, that is the in vitro model that they are using to prove the optical properties for imaging of this material. So they observe the internalization that is higher when they have a targeting molecule than without the targeting molecule that they are using. And in vivo is the same response, that they can have the magnetic imaging with in the presence of gadolinium when they are targeting the tumor cells. Just to have a brief overview, so depending of the properties of the nanomaterials, so we can have also fluorescence imaging. Then we have to take into consideration the depth of the unhealthy tissue to have a successful response of the modality that we are using. Sometimes we are developing, or there are some alternatives, like to use quantum dots, or as we saw, some of the lanthanides that have magnetic properties at the same time by its their electronic configuration will present other physical-chemical properties, such optical, let's say, such as fluorescent. So will depend of the composition, again, the chemical composition or at the same time, if we are labeling or we are encapsulated fluoroforms that we can follow in vivo for the imaging. So for this introductory part regarding in vivo imaging is the material that I prepared for you today. So if you have any comment regarding this topic or question, now is time to hear from you. Indeed, these kind of platforms are already in clinics, in advanced clinical trials. For instance, in France, there are a company developing or that have developed nanoparticles that have gadolinium and are used like a theragnostic platform. So they use the magnetic properties of gadolinium ions with the radio-enhancing properties of a high set atom that the gadolinium is. So it's really promising and a reality, the use of nanomaterials in theragnostic. Any comment?
Yeah, Eduardo have a comment in the chat that he mentioned, or he says that's beautiful imaging tools, definitely an option to pre-select patients for nanotherapies.
Thank you, Dr. Eduardo. So thank you, everyone. We can continue with the next topic. Thank you.
Okay, so two left. So let's continue with the following topic, the next topic, which is immunotherapy. Okay. So this is a quick outline of this part of the presentation for this topic. So I just will move to the basics of what cancer immunotherapy is, and of course it relies in our own immune system. So I guess everything starts with the release of antigens. Of course, antigens that are very specific to the disease. Those antigens go to the what is called antigen presenting cells, which one of the most common antigen presenting cells, IPC, are dendritic cells. These dendritic cells, they interact with T cells, mainly T cells, in do with the process what is called the priming and activation of T cells. When the T cells are activated, of course, the T cells, all these is, I guess the antigens starts or are generated by cancer cells in the tumor that encounter, as I was mentioning, the dendritic cells and in the lymph nodes, which is one of the main parts where this process of immune response happens, where the antigen presenting cells and T cells encounter the process of priming and activation. So the T cells go, yes, are transferred or are transported through the blood vessels to the part of infiltration into the tumors, and certainly one of the huge advantage of the response of, or at least this type of immune response from our bodies that is very, very specific. Once it gets into the tumor, it recognizes what is tumor cells through the antigen. So that was originally generated by the tumor cell, which is again very characteristic. Once that happens, those T cells recognize those cancer cells that are overexpressing that antigen, but the function, the work of the T cells is to get rid of, in this case, kill the cancer cells. So I guess one of the big questions in cancer biology, I guess in particular, is why our immune system hasn't been able to recognize cancer cells. Why we have that disease and the immune system is not able to pick it up. So there are different reasons. Of course, this comes through decades of research, through first to know in details how our immune system works, and also to know how cancer are I guess fooling the immune response, or cheating the immune response, either word that you want to use. At the end, it bypass. There are different I guess, or what we know around now, is there are different ways that cancer and the tumor microenvironment in cancer, which also is very important, to avoid, again, the work of the immune systems. Some of the tumors, some of the mechanism, they reduce, or they reduce the function of the T cells through different I guess that's the general outcome, to different mechanisms to avoid that function of the T cells. There are other ways to do that, secreting different signals that also bypass or hide the presence of those cancer cells. Like for instance, like the checkpoints, which are, there is a current therapy in the immune, or current immunotherapy, or current approach in immunotherapy that is pretty well used and known in the last few years. So the tumor microenvironment changes the expression of checking markers or checkpoints, and that also helps to the tumor avoid the function, or again, as we were saying, foolish the immune systems to avoid the killing of those cancer cells. So these are just some of the known mechanisms that tumor use, or tumor microenvironment use to avoid the immune system. So what are some of the current therapies that are used for immune therapy? And again, this is particular for cancer treatment. So dendritic cells, as we saw, the antigen presenting cells are very, very important. So in this case, the approach is to use dendritic cells, modify those dendritic cells with the antigen, very specific for the patient. So this is, again, a personalized approach where doctors, physicians get dendritic cells and antigens from the patient and are introduced back, I guess, after they are engineered in such a way that these dendrimers, not dendrimers, dendritic cells, sorry, carry the antigen, are injected back to the patient and do the cycle that I was mentioning in the first slide, second slide of the presentation, that once the antigens, the dendritic cells has the antigen, they will present to the T cells, and all the process of of getting rid of cancer will happen. Which I mean, in some diseases, in some type of cancer, it might happen. But again, there is not. I mean, the tumor has developed other approaches or other strategies to actually overcome this strategy of using dendritic cells. So if it's not, we will see that that cancer will be pretty much close to be cured, which is not the case. So again, it's not that simple. It's not that simple. I guess that's the message that I want to give him. In if this strategy of dendritic cells or T-cell therapy seems to be very very interesting, and since in principle, in theory, since could be successful, because at the end you are recruiting your immune system to do the work. That's what nature has worked in. I mean, in our own bodies for thousands of years to make our immune system very well, not perfect, but very efficient in terms of getting rid of foreign material or material that is not good for the body. But anyway, despite this seems very theoretically very important, or very that could be very successful, is not happening. Again, because other reasons. Another strategy is T-cell therapies. In these T-cell therapies, they are actually modifying the T cells with what is called chimeric antigen receptors, or some of you have heard about CAR T-cell approaches or therapies. And I guess in this case, they are actually going directly for the T cells instead of the dendritic cells, like the previous approach, where the dendritic cell still has to have one to go through one step, of priming and activating the T cells. In this strategy actually goes directly for the T cells and engineer the T cells with these chimeric antigen receptors. Again, there are the two different approaches. Actually, just bringing some of my collaborations, Dr. Mukherjee, she actually developed CAR T cells based on these tumor specific or tumors associated one that she developed at the beginning of her career. So she's loading these antigens based on that antibody to target these T cells. Again, and in principle, it has found that there are the pancreatic cancer cells overexpress those TIM1. So that's another technique. Again, these are general techniques. I still haven't touched nanomedicine yet for the application for immunotherapy. I just want to touch some of the basic points in immunotherapy. And the last one that I'm going to present is the checkpoint blockade. As I mentioned, one of the strategies for tumor microenvironment to overcome the function of the immune system to get rid of the cancer cells is overexpressing these checkpoints. So the idea is to blockade these checkpoints, and there are two that are very well known right now, and in testing in different trials. I mean, seems to be, I guess I'm over exaggerating, but pretty much in any clinical trial addressing cancer treatment with chemotherapy or other type of approaches, everybody want to put as an arm the use of this checkpoint blockade. And again, they are not perfect, work pretty well in some of the diseases, and that was the reason has been very used or are used, but it's still there is not a perfect solution. Okay, so what is the use of nanomedicine for immunotherapy, or what are the advantages of using the nanoparticles? Well, I guess everything goes back to the main principles. I guess where we start this tutorial, this session, in terms of the tunability of the nanoparticles, the size, starting for size, the chemistry that can be used, what can be loaded, how can they be modified. And for this particular topic, well, one of the advantages of nanoparticles is that you can either physically or chemically attach antigens to the nanoparticles. And also, well, antigens, and I guess just following the illustration, you can also add adjuvants to the same platform. So like we were talking, like we were talking in the previous presentation, Dr. Daniela was mentioning about theranostics. Here will be not only the presentation of the antigen, which will trigger the immunotherapy, but also some adjuvants, because sometimes the immune response is not as strong as we would like it to have a therapeutic response, so it needs adjuvants. And the beauty of nanoparticles is you can put both in the same platform. And the principle is I guess it's the same. The nanoparticles will go to the antigen presenting cells, release the antigen and the adjuvant, and well, those antigens will be presented and follow the cycle that I was mentioning before. So the dendritic cells, yeah, will prime, activate the cells, and so on, and we will follow the immune response to have for the treatment of cancer. There has been different platforms, approaches that has been used for to pursue this strategy of the use of nanoparticles for immunotherapy. And again, I mean, platforms pretty much is kind of go back to the list that I show you when I was describing different drug delivery system for cancer treatment, all that list of, I don't know, 10 platforms and more. So all those platforms that has been used for drug delivery system, at least what I have seen in the literature, people has been tried with more or less success to use for immunotherapy. In this particular case, is just again just an illustration, kind of the same message at the end of delivering actually peptides instead of the whole antigen that can be a protein, just delivering the active site, a peptide, and together with this particular case is a Freud's adjuvant, and well, they actually saw the enhancement on the therapeutic effect. And this is just an illustration of the platform, how it looks like and was modified with the peptide. For immunotherapy, it's pretty complicated to get reliable data in vitro. So most of the data is in vivo, so go to the animal model and treat it, challenge I guess the disease, challenge the animal with the vaccine. At the end is a vaccine, what we are developing here, and see if there is an effect in the tumor. And this is just again illustrating very well, making a comparison just when those peptides are just floating around and are presenting to the dendritic cells. So the effect of having the nanoparticles, the amount of peptides that can be released by just one nanoparticle, that will enhance the actual immune response, and of course the therapeutic effect at the end will be stronger, and you will see reduction in the tumor, higher survival of those animals that has been vaccinated with the platform. In this particular example, I'm bringing the use of actually mRNA to being the I guess therapeutically active or immunotherapy active molecule to actually produce the antigen. One is released at the inside of the dendritic cells or antigen presenting cells. So this is just another technology, and actually that's, I guess at the end of this presentation, I have one slide or two slides related to COVID-19. That's two of the vaccines. That's what they are doing, releasing mRNA on the dendritic cells, the antigen presenting cells, and following the machinery of the cells through mRNA, the proteins are produced, and those are the antigens that are presented of those dendritic cells. And again, it follows the cycle that I show in the case of COVID-19, of course, is to eliminate, or the COVID-19 vaccine, sorry, is to eliminate COVID-19. But in this particular case is for cancer. So these nanoparticles are believed are liposomes, or some sort of liposomes that are carrying the messenger RNA. That messenger RNA again is once is internalized in the cells, the platform is released. That's the ultimate goal inside the cells, and again the messenger RNA will be code to the corresponding protein, which is the antigen. And here is just a presentation of the results for this specific paper. I won't go in detail, but just mentioning that they actually observed that the platform was encapsulated or targeted in the spleen. Is another place where a lot of the T cells are all this machinery for the immune response happens, lymph nodes, spleen. So they saw that as a positive effect, and they actually saw the therapeutic effect in terms of producing more more immune signals for the treatment. I don't think I have but I don't have the survival, but I mean, based on the data, seems to be very promising for increasing the survivals of these animals. And finally, I just wanted to introduce, well, not introduce, but just to mention the impact of nanotechnology, nanomedicine in the last 2 years or so, when the pandemic started. Development of these vaccines using pretty much like technology that I show in this paper, in the sense of these are lipid nanoparticles, liposomes that are carrying mRNA that go to a specific or the dendritic cells again to activate the immune system. So something that we were discussing before launch is that I feel at least now the general public is more familiar with what nanoparticles is about. Most of the people in a positive way. There are some people that are having crazy ideas of the use of nanotechnology, of nanoparticles for vaccines, but anyway. But I think in general, this this is, I mean, it's unfortunately that something like a pandemic has to happen, but this is bringing good news or good marketing to nanomedicine and nanotechnology in general. At least that's my opinion. Okay, so that will be the, and this is just a description of Moderna. Actually, I was looking at the slides, and this before Moderna, the vaccine for Moderna was approved for clinical applications. It was in clinical trials, but anyway, that's what I have for this topic, immunotherapy. Does anybody has any question or comments about? Let's stop share.
Thank you very much, Juan. I think this was a very relevant part in the tutorial, because the same way that we were saying before, that is getting more and more complicated to get funding for nanomedicine in general. I think now if you include immuno-oncology in any of your applications, then your chances increase automatically. So I think this is becoming very, very relevant. I want to ask you, because now after this lecture, I just remember one of the first comments that you made this day about your colleague, Professor Afonin, and then some immune effects with siRNA delivery, if I remember right. I just wanted to know if that's a good effect or a bad effect in the immune system, and if that was provoked by the nanoparticles or by the treatment itself, the RNA interference. I guess we will have more about that tomorrow, but I just wanted to have an idea of what was that about.
Yeah, let me see if I got your question. So the question is about where the immune response due to the siRNA nanoparticles come from. That's kind of your question, yeah?
Yeah, that's it.
Yes, okay. So, I mean, RNA, DNA in general, cells, body has a specific, well, let's put it, cells has a specific mechanisms to recognize, right? Because the cells doesn't want foreign DNA, RNA to go inside, right? Because it will impact the original genome of the cell. So there are specific mechanisms to recognize that. So it was a direct, as far as I understand, it was let me put it back. His research is trying to overcome some of the issues that people in his field, siRNA delivery, has found. So there were clinical trials where they delivering siRNA and using, I'm not sure about the platform that they were using, but it created immune cascade, I think they call it. It triggered an immune cascade response in the patients that I'm not sure if even one of or some of the patients died because of that. They went back and they found out that the immune response is due to the siRNA. And again, it's because cells or body doesn't want to have that. They are mechanisms to protect of that, because that will be complicated, right? If we have every time foreign RNA, DNA getting in our cells, changing our own genome. That's my understanding. So what he's doing is actually developing siRNA nanoparticles, or nanoparticles based on siRNA, put in other ways, that seems to be that they don't have that kind of response, or the response is lesser than just as siRNA by itself. But I think that was a little bit more of what you asked for, but yeah. But anyway, that was a natural response from the body due to.
I see. Okay. Thanks so much.
Thank you. Okay, what I did. Okay, so the last one. So this is the last presentation. Okay, so the last one deals with a little bit looking at the, well, I guess exploring what we have right now in terms of the use of nanomedicine or nanotechnology for biomedical applications. Exploring a bit of the challenges, more general challenges other than scientific challenges. I will still mention some of them, but that has prevented some of these platforms, at least these platforms, to move forward. And as Eduardo mentioned, at least the field or some people in the fields things that and partially agree. I be honest, maybe not totally agree. I think it was more, as I mentioned, the field promised too much from the very beginning. That's what seems to be looks like a failure. But I partially agree that there are not that many nanoparticles platforms used in clinical applications as it should be. And there are a lot of factors. I mean, together with promise too much, there were platform projects that were looking in the wrong directions or looking in the wrong animal models or looking in, anyway, there are different factors. It's hard to bring all of them there, but the reality is very low number of platforms. Considering the effort, considering the millions of dollars, I mean, just to mention US as an example, the nanotechnology or the biggest project in nanotechnology for US started in 2000, so it's around 20 years, and in particular for cancer treatment, NCI, started in 2005 or so. With all these cancer centers and that. So a lot of effort, a lot of millions, billions of dollars. And at the end, we just have, I guess, very few platforms there in the clinic. I don't know, 20, 30 platforms. And it's the same for other, the use of this platform for other diseases. And this is just a graph that I put in here in the presentation that show the progress of different platforms that has been approved based on nanotechnology and for very different variety of diseases. This is in particular for cancer. I mean, we have this is data from 2019. We have almost 200 clinical trials in phase 1, phase 2, phase 3. You can see that it goes down very dramatically from 91 in phase 1 to phase 3, to only 21 clinical trials. So the success is the rate of success of starting in a platform, starting with a clinical trials phase 1 to phase 3 is pretty low. Actually, I guess that's the number, 14%. That will be the success. These are just different reasons why some of these nanomedicines has failed, some of these platforms. Some of them has to be with with the toxicity associated with those platforms. Could be immune response. I'm not sure in these specific examples. Right now, I think with what we have learned in all these years, that's not that very common in phase 1. I think with all this characterization that I was mentioning before, all this cascade of techniques that we have to characterize in vitro, in vivo, I think we're in a much better point to to determine whether a platform should go for phase 1 clinical trial or not. So usually these days, a platform failing phase 1 is not that common. And you saw in the numbers in the previous slide, actually the clinical trials phase 1 are kind of high, 90, almost 100 there. Phase 2, one of the, I guess the main issues with phase 2 for these platforms to fail is that they miss the primary endpoints. Primary endpoints usually deals, at least for cancer, deals with improving the efficacy in terms of the treatment of the disease, which they usually measure in just in a simplistic way in the months of what the patient will survive for their specific conditions. So if the current treatment, statistically speaking, will say that the patient with that disease will survive, I don't know, 5 months, and if by using the nanoparticle version of that treatment, the patient will survive 6 months, 7 months. That's what will put the platform forward for phase 3, or will say it won't it won't go, or it won't move forward. And with a lot of these platforms that fail phase 2, that's what happened. Either didn't, or either they just were statistically similar to the actual treatment, or if there was some improvement, was a matter of days. Or there were cases where actually the use of the nanoparticles was even worse than the current treatment. And in phase 3 is also kind of the same issue, is failing the primary endpoints. Of course, as you might know, again, with the vaccines that has been developed for COVID-19, I think as a general public, we are more familiar with clinical trials, what phase 1, phase 2, phase 3 implies. Usually from phase 2, phase 3, of course, there's going to be specifics for the clinical trial, but in general, it's just having more people in phase 3 instead of having 100 or 200 as they might have in phase 2. In phase 3, we're talking about thousands of patients that are treated, and that's I guess more challenging to observe some response because again you have more patients there. Or at least that's what we have found, that sometimes that platforms that do well, okay, in phase 2, phase 3, they just fail because, again, statistically they don't see it to improve much of the treatment. Let me just point out here, and again, sometimes comes ideas to my mind, like sometimes the treatment or improvement of the treatment might not be the ultimate outcome, or might not be that priority. Has happened with Doxil. Actually, Doxil, which is the platform that I have mentioned in the previous topics, that Doxil actually didn't improve the treatment of the disease. Actually, the reason why Doxil was approved at the end to be used in clinic was because it reduced dramatically the side effects of the original drug, which is doxorubicin. So sometimes some of the platform are that's the way that they get into clinic because of the safety or reduce of the side effects. I just put this as an example of a platform that failed, and at least for the field of nanomedicine was a huge failure. This being O14 on Professor Langer and MIT that was developed from his lab was a big hope for us in nanomedicine, that this will be the great tool or the great weapon from the point of view of nanotechnology to treat cancer. At least that's what I remember, that we saw this nano platform 5-6 years ago. So at the end it was, I mean, I remember seeing the platform used for this cancer, other cancer, and some other cancer. I mean, they were trying this in 3 or 4 or 5 different types of cancer, with, again, with the promise that it will be successful. But it seems to be that in phase 2 clinical trials, the platform didn't reach the primary goals, and this is the platform that in some of the cases actually did worse than the current treatment. So certainly, I will talk a little bit about more of related to money in some of the other slides, but at this point of phase 1, well, I guess just from phase 1, certainly phase 2, money is a big player, because we are talking about clinical trials that cost millions of dollars. So no university by itself or institute by itself can pay for that. Here is we need to involve, or people that is running these clinical trials need to involve investors, private companies. So now is the games is of course is still scientific, is still thinking in cure the disease or treat the disease, but also business starts to play a role. So if the investors didn't saw a very good outcomes of the, or actually did not see a good outcomes from this platform, but they start to take their money, and at the end the company went bankruptcy. So was a huge I guess bad news. And I remember this was, I think it was like 4 or 5 years ago, and I as I was mentioning, the nanotechnology or nanomedicine was already going down, that people start to look with more detail, is what they are promising in the proposal is really what they are bringing. And now with this news, people even say, well, it's a completely failure, this field of nanomedicine, and shouldn't even exist. I remember seeing an editorial in the Journal of Control Release kind of using those terms. Anyway, so there are three main or broad factors that explain the failure of these platforms. Science, some of the topics for science we have already gone through, so I will go quickly. Clinic and the market. Well, science, as I was mentioning, well, it's very important to understand the characterization of those nanoparticles and understanding how it works in the biological systems. I mentioned this quote that before we were designing nanoparticles against biology, now we are designing nanoparticles with biology. We'll try to take advantage of that, as I mentioned in the protein corona. But instead of trying to get rid of the protein corona, let's try to use in favor production, these good manufacturing practice standards. We as a scientist, well, we have our lab, we have some beakers, put together certain compounds and make our nanoparticles, and that's it, and we have our nanoparticles. So that's not how industry does their work or make their materials. And one of the requirements, like for instance, for going, as far as I know, going to or to apply to go for phase 1 clinical trials, you have to prove that you are bringing your platform to, for that application. You have to prove that you can make your platform using these manufacturing practice techniques that imply a lot of different protocols to make sure that starting with your materials that they are not contaminated, the certain grade of purity, and all the protocols that you use in the lab to synthesize those techniques. Anyway, I won't want that in a level, too much level of details. The point is very critical for us to demonstrate, and it's complicated based on the culture that we have scientific labs. We usually don't care, or at least, if you see a chemistry lab, it's usually kind of dirty there, that kind of things. But there are actually companies that help to with that. If you have the money, of course, or if somebody is really interested in the platform, that will help to translate that production of the platforms using these standards. Clinical safety issues, as I say, might not be that much as a problem, but there are still can be some issues. More so a problem, again, if you are approved for phase 1 clinical trial, most likely the FDA, which is in US, the body that regulates those approvals, most likely will have enough information to kind of say that, at least from the toxic point of view, shouldn't be that a big problem. EPR effect, well, I think we already discussed a lot about that. Pharmacokinetics, again, we are still dealing with the immune system, which is the translated, or in other words, the mononuclear phagocytic system, or RES, that we used to call it in all times, is still taking our nanoparticles from the body and clearing there, with having much efficacy and in accumulation in tumor. This was another big bad news for the field when this review come out saying that, analyzing a bunch of papers in the literature during 10 years, seems to be that the percentage of nanoparticles that actually reach the tumor was below 1%. That was very shocking for the field, hearing those numbers. And something that I also mentioned during this presentation is our animal models. The easy one, the Synograph, that is easy to work with, cheap, is we cannot rely. It still might give us some information, but it shouldn't be the last proof before moving to approval for clinical trials to move our platform for that. We should use other, a little bit more, more developed animal models that resemble better the actual disease that is targeted with that platform. Clinical failures. Here the challenges are, I guess, different sometimes. What happens is we have our idea from the chemistry point of view, materials point of view, and we know that idea works very well with a specific drug, and the nanoparticle is beautiful, is perfect, and we want to push that nanoparticle for that, with that drug, to try to cure, to treat a disease that in clinical, in the reality, or in clinic, that drug is not used. So that's what happened to this platform that I was mentioning before. They tried to push it to a disease that, again, that was not the primary care that drug was not the primary care for or the primary treatment for that disease. So, so they think that was one of the reasons also why the platform failed in phase 2. The right patient selection is critical to successful clinical trials. Well, one idea is, as we were mentioning with the APR effect, well, is not every disease has APR effect in the same degree, not every patient, so as to make it more personalized. So in principle, that will be the ideal route to go, to try to test or pre-test the patients and see if they are suitable for that. That actually is also expensive, try to do that. And, and also what it has happened with a lot of platforms that, I mean, these are new approaches to treat cancer, where people might not want to risk much, either investors or even the agency, the government agency. So usually a lot of these platforms are tested with patients that are in the last stage of the cancer, which is, I mean, the most challenging stages to treat. So they are put or try to be tested or proved at those with those patients, which is complicated. I mean, even platforms that might have some promise for the disease with other type of patients, patients, sorry, at other stage for the same disease, we don't know, because again, those are tested with the ultimate type of patients, or that unfortunately they might not have any other hope to be treated. So combination is very important. I think it's idea that is bringing more, I guess, track to the field, that now treating cancer with one drug might not be the right approach, maybe combining drug, but that's still complicated for the nanomedicines, because again, we need to make sure that we have the right combination. For instance, I put as an example a work from my group where we combine cisplatin and gemcitabine, but I'm being honest, that combination cisplatin gemcitabine is not currently used to treat pancreatic cancer. Our hope is that our work will attract people's attention, is because the reason why cisplatin gemcitabine is not used to treat pancreatic cancer patients is because cisplatin is very toxic. So by having cisplatin the nanoparticle, we're hoping that toxicity will be reduced. But again, even it seems to be a good combination, yeah, is clinically speaking might not make more sense. Yeah, so even you have positive data or positive outcome at the end of your phase 2, phase 3 clinical trials, sometimes it happens that from the regulatory point of view, maybe the protocol or the clinical trial was not doing correctly, and this deals more with the protocols, and as mentioned, I mean, I have never submitted a clinical trial, so I'm not that familiar with these specifics, but what is mentioned in the review, where I'm taking some of the information is yeah, sometimes people doesn't have the experience and design the clinical trial in such a way that seems to be that the government agency, FDA, I guess mainly in US, just because it was done that way, I guess maybe don't have the right controls or enough controls, is not approved. Here I would like just to review I guess the different steps that we need to go through to develop a platform. move from what we call, as we say, scratch, starting in the lab, all the way to put that platform in the market. And I guess one of the main things that I want to make sense for I guess for everybody in the audience is, I guess, the time and the resources that is needed. So we usually start in the lab with an idea. We start to test that idea, synthesizing the nanoparticle, characterization. That's what we mentioned of that, of physicochemical was have been mentioned in this tutorial. Different type of techniques, in vitro and in vivo studies. Scale up, as I was mentioned, has to comply with very specific manufacturing standards. And this is another scheme, I guess, showing the part of the clinical and also the commercialization. So an idea starting from the lab can take 5 years or more to move it to all the way to in vivo. That's a lot of resources already. I mean, if I can tell you at least what I have worked with some of our platforms in the lab, we had got to in vivo evaluation with some promising data, and promising depending who, because if you ask the reviewers of some of the proposals, they might say no, not that promising. But anyway, we haven't even touched the part of the scale up the material to comply with this manufacture regulations. And from there, and there I'm talking about almost 10 years working on that. And from there, of course, has the phase 1, phase 2, all these clinical studies, clinical trials that can take from 1 year to 7 years. I mean, just to bring in the vaccine as an example, it took 2 years for those vaccines to get approved and commercialized. And a lot of us, we actually have that vaccine in our body. But also what is important is again, like the patients, the number of patients that you need to involve on those clinical trials, and also and from there, once it's approved, well, you just start the process of commercialization of that. Anyway, I was looking for other slide. Maybe I didn't include it, but what the other slide was mentioning, and it might be in the wording in the next slide, but was mentioned that these clinical studies require millions of dollars for that to happen. So that's something that, again, a university, or I mean, there is not, as far as I understand, there is not a specific grant from the US government that will say, well, I want to support you with all these different clinical studies for you to, I mean, there is not, we are talking about millions of dollars, tens of millions of dollars for those studies. Now, there is the other perspective, and here now I'm going to start to involve more money. Or no more money, but talk about more about money, is okay. So we have our technology approved, it's being commercialized. So how much it costs any of these nanoparticles that are used? And I have an example from Abrasion. So Doxil was the first nanomedicine approved in the 90s, or 1995 actually. Abrasion followed that. I think it was 2000 or 2005. I don't remember exactly the days. Anyway, that was the next one to be approved, or at least in US. And that Abrasion carries paclitaxel, and is currently sold in, I think the market is hundreds of millions of dollars. That's the market for that product. But I guess what is interesting here, or at least from my point of view, a dose of Abrasion costs $10,000. Compared with the cost of the paclitaxel, which is the drug again, which is $200. Now, we're talking about 100 times more the cost of that, and unfortunately, and that's one of the, I guess, some of these numbers that I'm bringing here. Some are the numbers that investors or companies look at when they say, well, we passed phase 3 clinical trials, but what is my market? Is my customers gonna pay $10,000? Is if the Abrasion, instead of giving an average survival of 5.5 months, is giving them 6.5 months. And of course, I understand, I mean, we're talking about people, that's the ethical, the other side, right? If we are talking about the ethical side of this issue. But anyway, as companies, investors, they might say, well, no, the market is not big enough. Let's just discontinue this technology, even if I mean, it was successful, it treated the disease. Again, was not giving twice the enhancement on the survival, but it was being promising. But maybe the company can say no, it's not a good market. So that could be another reason why these platforms has failed. Yeah, and just to bring, oops, again, I pushed the wrong button. Just to bring another that could be an ethical topic is how much it costs for a company to produce a nanotechnology. And unfortunately, just to have the full disclosure, I don't have the numbers, but I found how much it costs to develop a new drug. And it's 2.5 billions. Starting from scratch, from the beginning, or 1.5 billions, depending on where you look at. And I don't think nanoparticle will be cheaper than that. So again, and I guess the ethical part, what I was bringing, and that's just I guess information when we were talking about patents, to release the patents for these vaccines. I don't know, the companies already invested billions of dollars. I'm not sure they are willing to release those patents. Anyway, again, here we are talking about business. It's not much science, but also impact the success of these technologies. That's what I bring in here to the conversation, or presenting in this tutorial. On the same topic I bring to my students when I give this class, because we also need to think about that. Well, again, some of the topics for market I already mentioned. Maybe not enough funding. I mean, the cost of those clinical trials might be are too expensive, and the investors are not willing to pay if the nanoparticle didn't, the results, let's say for phase 2, were not that good. So they just say, no, I'm not ready to invest another $10 million just to maybe say, well, that's not, it will not gonna work. We are not gonna prove that. The second bullet point is pretty much the question that I was making, the ethical question, or is really worth it to spend 100 times to pay for something 100 times more than is just gonna give a few months or a few weeks of life to the patient. And there are other, I mean, nanotechnology, nanomedicine are not only the only therapeutic agents that are being developed to treat cancer or other diseases. There are a bunch of other technologies out there. So how competitive are these technologies or nanomedicines compared with all those technologies. And competitive, here again, we are talking about money, markets, yeah. Yeah, I mean, having said, yeah, these are the last slides. I mean, having said that there are failures, that there are a lot of challenges ahead of in the field, and I mean, I anticipate, and I'm not the only one anticipating that it will grow. It will keep growing. I think from the business point of view, just bringing again the example of the vaccines for COVID-19, from the business point of view, I can see that a lot of companies that were not, that they might bought some startup companies 5 years ago that has some nanotechnology there and they were not looking at. I can guarantee that those business people now is looking at, oh, let's bring back this startup and let's see what we can do and what we can move forward, because they start to see money that that these technologies can bring them money. So yeah, so I anticipate that the nanomedicine field will keep growing, at least from that point of view. I mean, that some of the, liposomes, I'm pretty sure that there is now, from, again, the market point of view or an industry point of view, yeah, a lot of eyes put in liposomes. That's, something that are already, that will be helpful or not for other platforms, like, in my case, other groups that are working with different kind of platforms, that I'm not sure. But let's see. So yeah, so I think that's the last slide that I have for this topic. Any question or comment?
There is a comment from Dr. Eduardo. Patient selection is the way to go, in my opinion, but will indeed make cancer nanomedicine a very expensive treatment.
Yeah, that's certainly, I'd say certainly for sure, because right now, well, I'm speaking based on what I understand. I guess the model is to develop a medicine cure that will help a bunch of people at the same time, and that will reduce the cost of the medicine, or at least that's the idea. When we're talking about personalized medicine, is for you to have a pre-test to find your, I don't know, antigens. I just throw in some words there, antigens, or find if you have EPR effect in your disease, your body, and for you to take those antigens and put it in a specific nanoparticle that is going to be specific for you. Who is going to pay for that? You. So that's going to be expensive. But no, anyway, personalized medicine is a big boom now, also in nanomedicine and other fields. And maybe at the end, that's what it will take to treat these diseases. If we are talking about cure, I will say, yeah, if we are talking about extending life, well, for a few months, maybe that's too expensive. But if we're talking about cure, I think that's the way to go, in my low or very very short understanding of this. Thanks for the comment, Eduardo. Okay, any question? I mean, I seen some comments, and I really appreciate your comments. I think it was fantastic. It was, I guess, challenging for you, challenging for us in terms of the time was kind of long. But yeah, we really appreciate that you stay with us and your comments or your questions. I'm not sure if you want to say something or no.
I will just say thank you to everyone. For me, it was also a really nice experience. So I don't know if from the conference organizers there are any
Yes.
comment.
Yes. Thank you very much, doctors. It has been a very wide and very enriching information. So let me share my screen, please. So on behalf of the Mexican Materials Society, I deliver the certificate to Dr. Juan Vivero Escoto for providing the tutorial Nanomedicine Principles and Applications, and the same way to Dr. Daniela Edith Salado. It has been a very enriching tutorial. So thank you very much, doctors, and to the assistants. The list has already has taken in the session. In the certificates will be mailed to all. Thank you very much and have a good evening.
Thank you.
Thank you.
To see you tomorrow in the symposium.